Right, I think that we've reached that point where we're going to get started. So a very warm welcome to you to this this COG-UK showcase event today. It's our first showcase event that we put on to date. We've done several seminars in the past, but this is our first showcase event. And I'm delighted to offer a warm welcome to you. If you don't know me, I'm Sharon Peacock. I'm the director of this consortium. So I want to really only spend a few minutes largely saying thank you to people. And so we have a fantastic lineup of speakers and chairs today. So a sincere thank you to everybody who's agreed to do that for us. The lineup is grouped into 3 sessions, which covers different topics. And you'll see that you do get a break in between the 3 sessions. And so you just get a chance to catch your breath. And we will be asking you to put questions in the chat, please, and we will pick up those questions in the chat. So Andy Jeremy, who's helping us to administer the meeting, will be able to pick those up, and we can direct those to people as and when we have time to ask them, although it is quite a packed agenda. Now, one of the things that's been on everybody's lips in the last few days is the new variant of SARS-CoV-2, and we have asked one of our speakers to include some details of this. So Eric Volz at 4:30 will be talking about that. Now, if you don't manage to stay for the duration, this whole program is recorded, and so you'll be able to go back and listen to that and catch up later. But the only other thing I would say is usual Zoom etiquette. If you could stay on mute, we will actually be muting you centrally. So if you want to say anything, you will just have to unmute in order to be able to speak. So without any further ado, I will actually hand over to Sir Patrick Vallance, who I have to say has been a tremendous support to us from the very outset. So, you know, this has happened really through the enthusiasm of both the people in the consortium, but also people in government, and Sir Patrick has been a huge supporter of us from the outset. I'm sure that his enthusiasm was one of the key reasons why we've been able to do what we've done. So thanks to him. And Sir Patrick, you're just going to give us a few opening words before we go on to session 1. So thanks very much.
Yeah, hi everybody, and thanks Sharon. I looked at your agenda this afternoon. It looks absolutely brilliant. And I think about what I've got to do this afternoon, and I'd really like to swap. So if anyone would care to do something else for the afternoon, just drop me an email and we'll have a swap. I went back to have a look at the initial emails around this. I was just trying to track them back in February, Sharon, when we were emailing about the need for this. And it's amazing to see how quickly this was set up, with more than 300 people in it now, and the links that you've got to the human genetic side of it with genomic, CIC and genotype to phenotype and all those sorts of things as well. It's incredibly impressive. And it's an area that the UK is genuinely world class at. I mean, it's something that we do very well, you do very well. I think it's shown its worth already, and has been very, very relevant to how we think about this epidemic, whether that's the history that you've been able to show, in other words, the seeding events that took place in the UK in February and March and how that happened, or the ability to track and identify where clusters have originated from, and indeed things like working out whether spillover from university infections through into communities is occurring or not. All of those areas I think are incredibly important and relevant to how we think about managing this. And as Sharon has already said, the rapid identification of new variants that may or may not have different characteristics, I think is going to be very important going forward. So I think it's been impressive. I'm incredibly grateful to all of you actually, because I know how busy everyone is, and that almost everybody is just doing this as well as everything else that you're already doing. So it's impressive and important. And I think it also really should and must define how we think about infection management and tracking and tracing going forward. I mean, this has to be the way we do things, whether it's in foodborne illnesses or in respiratory illnesses or anything else. I mean, this has got to be the way that we start to get a real handle on this. So I think as always, a crisis has prompted an activity which I think many of you would have thought was necessary anyway. And hopefully we can now make sure that we get this really embedded in the system going forward. So I hope you have a brilliant afternoon, actually, of fascinating science. And as I say, I wish I could stay to hear all of it, but I can only stay for a short period. But thank you once again from me.
Patrick, that's amazing. Thank you so much for that endorsement. We really appreciate that. And if you want to go back, then we can make the recording available. So I think that we should move on then to session one. And what I'm going to do is hand over to Dr. Rob Orford, who's the Chief Science Advisor for Health in the Welsh Government. And he's going to be chairing a session on the overview of lineage, introduction and transmission in Scotland, Wales and Northern Ireland. And then Professor Deenan Pillay from UCL is going to be trying to draw together the strands from that to think about the implications for policy and for practice. Now, most people are showing their own slides. I hope that's going to work out well for us. If people have glitches, we've got a system in place whereby we can just go on to the next talk if you're having problems and then go back to you. But hopefully we'll have a smooth run through. And so we'll buckle our seats, and I'm going to hand over to Rob now. So Rob, I'm hoping that you're online. Thanks very much. Thank you.
Thank you very much, and Sharon, and you know, some excellent reflections there from Sir Patrick. It's an enormous privilege to be with you. You know, you were giants in your field. You are making leaps and really furthering our understanding of the epidemic. And rather than having that retrospective look and learning, you know, we're using your information in real time to inform our response and our policies. And I do think this is the birth, or certainly the mainstreaming of precision public health. And I sincerely hope we don't go back from here. There's a lot that we need to do going forward, and there's enormous strength and power in the information that you are able to provide with the technique. So I'm not going to talk for long. I'm here to listen to you. There is so much fantastic work going on. It's going to be very, very difficult to contain these presentations in 10 minutes, but I will do my level best to chair over Zoom to help colleagues do so. So without further ado, it'd be great if Matt Holden can talk about experiences in Scotland for the overview of new lineages and transmission. Thank you.
Hopefully that's showing on the screen, fingers crossed.
Yes, it is. Thanks, Matt.
Brilliant. Okay, thank you very much. I'm going to start off with, I think probably, hopefully if the slides will move. The acknowledgements, which I think is a key part of the work that we've done, just reflects the fact that with the work I'm going to present, it represents a team effort, and this has come about because of COG. We're very fortunate in Scotland to have such experts in virology and genomics, and the partnership that we've built with the CVR, especially Emma, David and Anna at the team there, and also Andy Rambo's team in Edinburgh has facilitated this. It's brought together Public Health Scotland and also NHS partners. And really, I think COG has shown what you can do. In many ways, COVID-19's rewritten the rule book in terms of what you can achieve and get done. And really, COG has been a platform for introducing genomics into the response and actually informing public health measures. So I think it really is a template. And in Scotland, we've been very fortunate to be part of this. In terms of the sort of work that we've done, in Public Health Scotland, we've integrated the data within the organisation and linked it up to patient information that's allowed us to drill down on the detail. So that's been used to inform the response, but also to provide a surveillance backdrop, but also to target for outbreak detection and incident investigation, which again is helping us on a day-to-day basis. If we look at what's happened in Scotland over the sort of last 9 months, again, this is the epidemic curve that we've had. We can see we've had the two waves, and again, this has coincided with interventions that have in the first instance have been very effective, and we're now in the second wave. But genomics has allowed us to basically look under the curve into the events that drive this to get a greater understanding and extract from this learnings that will hopefully allow us to get ahead of the curve in future for this epidemic, but also more widely as we use genome sequencing as part of an integrated response to pathogens. And so what I'm going to do is just present some work that has been driven by the sort of collaborating partners with in Scotland with PHS that's provided some insights into that. The first sort of observation was to look at the sort of first epidemic and discover that the population that was driving this was diverse and arose from repeated introductions that then followed on from community spread. So this is a work that was driven by Emma Thomson, the team at CVR, along with partners in Edinburgh, so Kate Templeton and Andy Rambo and partners at Health Protection Scotland. And this showed in detail what happened in the first month of the epidemic in Scotland. And this showed that in the first case, the early cases had travel links with them, and in many cases back to mainland Europe. And the first couple of weeks of the epidemic, so at the beginning of March, many of those cases had travel links to them. And then over the following weeks, and by week 4, the majority of the cases had moved from those that had direct travel links through to no travel links, suggesting that the seeding of those infections then was leading to further community spread, and so on. This is the sort of set the scene for the rest of the epidemic, the introductions, and then more extensive community spread. And I'm sure that this story will echo in many of the other talks that we see in this session. And then we asked the question, what happened next in terms of the epidemic? As you know, we went into lockdown. And again, the analysis we've done has showed that the stringent measures that were put in place were very effective in not only reducing cases, but also extinguishing those viral lineages that had become successful. And again, reflecting on what happened in Scotland, we can see here as over a period of time, we've got a plot of the overall diversity of the lineages color-coded by the UK lineages. And we can see as we went through the weeks in the beginning of the epidemic through to lockdown, we can see the sort of diversity increasing. And we have a plot here that shows the sort of total number of lineages observed in an epi week. And after lockdown, we can see this tapers away as the diversity of the population that's circulating decreases as the cases go down. So we can see that the sort of measurable effect that lockdown had in terms of constraining the virus. And then again, to look in more detail at what happened, this is work that Sam Lysett has led with David Robertson over at Glasgow, a more recent study that's went as a SAGE report that's now on the COG Consortium website, that we looked in detail and we're able to break this down at what happened at the local level rather than just the national level. And in this case, looking across the health boards in Scotland, we could see the sort of effect that lockdown had, that after lockdown, certainly at the board level, there were immediate changes in the diversity of the population that we were seeing in the sort of more rural boards. And then eventually we also saw this moving through into the more populous boards, so the larger health boards such as Greater Glasgow and Clyde and Lothian, again, reflecting the observations that the sort of spread in populated areas was a key driver of this. But and also the impact that lockdown was slightly slower in those more populated areas where there was a higher density of population and higher incidences of disease. So again, it provides key evidence to show what happened across Scotland in the different health boards that we have here. And then moving on to what happened next, as we know, there was a lull in the summer, and then we've seen the appearance of the second wave in Scotland. In this case, we could see that in the second wave, that was a very distinct population that moved in. And again, this is a work from Sam's paper that shows the lineages that were circulating very early on, the ones that in green that appeared and then became extinct, the ones in orange are the ones that carried over into the second wave. And then blue are all the new lineages that started to appear and then become sort of more prevalent in proportion. Again, we can see where this fits in relation to the phases where lockdown was introduced and then we relaxed as we saw the disappearance of cases. And then after that period in time, invariably, we start to see the sort of increase in cases. And many of this has been associated with the sort of international travel, and again, reimportation of cases back as people then sort of resume their sort of normal lives after lockdown was eased and people went on holiday. We can see the sort of impact that this had. And again, this is just a plot that shows the composition of the top 40 lineages we saw in Scotland during the first wave, as opposed to the second wave, by epi weeks along the bottom. Each dot represents the relative amounts of samples of that lineage. And you can see roughly a pattern where the second wave are distinct from the first wave, in that you can't see dots under the first wave for many of the dominant lineages. And indeed, when we look at where the origins of those samples were and go back and trace through the epidemiological records for travel links, in many cases, those new introductions can be traced back to travel links back to Europe, and in this case, Spain, Greece, Turkey, and Croatia. Indeed, we have some evidence of some of the lineages coming from England as well, and some of these are probably reintroductions of lineages that we'd seen before coming back, and some of these were linked to cross-border outbreaks that we saw. And indeed, we also had some lineages that, again, we didn't have the epidemiological links to provide evidence of travel with key index cases, but in this case, the genome information and the phylogenetic reconstruction was pointing towards a distant ancestry beyond lineages that have been seen elsewhere in the UK and Europe. And again, in some cases, some of those dominant lineages were ones that we saw that were associated with some of the outbreaks very early on as the second wave took off. In this case, an example of how we were able to trace cases from Aberdeen that led to a lockdown on this long branch here that distinguishes it from the rest of the population, that started in Grampian and was associated with spreading in and around the city, that moved to the Shires and then elsewhere out on fishing boats and fishing crews to the outer islands, and indeed across to the rest of Scotland, that we're now seeing this lineage picked up in other outbreaks that we've traced across Scotland. And indeed, again, if we use the data, and this is the microreact tool that David Arnson and the team have developed and used, that's a very good interface for showing this. We can see the Scottish population here of this lineage very early on in the outbreak. It spreads around Scotland and very quickly spreads elsewhere in Scotland, and indeed throughout the rest of the UK, just showing the importance of being able to track these strains, but also to introduce interventions that can stop them spreading. And so in this case, in Scotland, the ability to sequence samples readily to identify outbreaks and also sort of target interventions is very important. One of the developments and one of the benefits of the COG partnership is that we've been able to set up and obtain funding from the Scottish Government for a rapid sequencing service that can just do this in terms of sequencing samples and providing information back to public health and infection control teams that allow us to target outbreaks effectively and be prepared for the next wave and other sort of interventions in the future when we approach the sort of programs such as vaccination and targeted interventions in response to variants and mutants. So I'm just going to finish there. Again, just to acknowledge the team that's brought this, especially the team at the CVR with Emma and David and Anna, the team in Edinburgh with Andy Rambo, but key partners in the NHS, Kate Templeton over in Lothian and Rory Gunson over in the NHS, and also the team at Public Health Scotland who've built this service and integrated it, especially Sharif and Kath Arnold and Michael Lockhart, and then also David Arnson for the work on microreact and the COG partnership for building this. Thank you very much.
Thank you very much, Matt. I'll wait for the applause to die down, but I mean, that was incredibly presented, so much rich information, so much work going on. So thank you for keeping to time. Just some gentle housekeeping. We're not expecting a fire alarm this afternoon. So if you get one, I would recommend you escape to your emergency rendezvous point. I'm not sure if we've had any information on the conference meal as yet, but I'll try and find that for you. Just for presenters, if you can put your full slides up, we'll be able to see more of your presentation. So if I can ask Professor Tom Connor to make his way to the lectern, I'd be very grateful. Thank you.
Thank you, Rob. So I'm going to give you a bit of an introduction into well, it's really a look back on some of the highlights over the year of some of the work that we've done. And like Matt, I'm going to be starting off with acknowledgements. And we have been fantastically fortunate in Wales to have a brilliant team associated with the pathogen genomics work. So in Wales, we have a pre-existing pathogen genomics unit with an extremely talented lab and bioinformatics teams who've really been fundamental, as you'll see in a moment, to getting this set up and running extremely quickly. We also benefited from a whole slew of collaborators across COG-UK, some of who I've highlighted here, without whom, a lot of what I'm about to talk about wouldn't really be possible. The sheer collective endeavour that COG-UK represents, I think, is absolutely phenomenal. I've never seen anything like it in science, certainly in the UK before, and it's been an incredible few months. So I thought I'd just start with a little perspective on kind of where we've come from in Wales and where we are now. So we sequenced our first genome, SARS-CoV-2 genome on the 6th of March, and since then we've really scaled up. So by the end of April, we sequenced 1,000 of those genomes. We had our genomic outbreak analysis and support routine service effectively in operation by the end of June. We'd already produced outbreak analysis reports. We produced our 10th outbreak analysis output by the end of July, and that sort of carried on, and we have a whole host of ongoing outbreak investigations that we're contributing to at the moment. In August, we were able to develop a set of genomic indicators which feed directly into the internal Welsh Government dashboards. This is providing some sort of summary information from the genomes that we're generating, which feed into indicators and circuit breakers that can be used by policymakers. At the same time that Scotland were looking at imports into following the first wave over the summer, we were also looking at that, and we generated a report on imports and reintroductions for Welsh Government in October, which I'm going to mention. And actually today we passed the milestone of 15,000 genomes sequenced by Pengu alone, which is incredible for a small team to have gone from from March. Starting our first one and then actually having 15,000 sequenced now, it's phenomenal. Even more so when you think that the standard sort of throughput we would expect on the upper end would be 10,000 samples in total from our clinical services in a year within pathogen genomics. So it's a real testament to the team as to what we've managed to achieve. And we're using that data at a lot of different levels. So we have outbreak analysis, which is provided at a very local level. And we've got a system in place that connects the bioinformatics and the lab teams up with healthcare epidemiologists and other colleagues across Wales to provide sort of responsive support for analysing outbreaks as they happen. That's sort of our routine service at the moment, if you like. We're also looking at national transmission. So in the middle is an image, and the size of the square, the colour of the squares. So squares which are blacker have more transmission locally. So this is analysis looking at the balance of where transmission happens, and that demonstrated to us relatively clearly that a lot of the transmission is happening in more urban areas, and the rural areas, if you get COVID if you had COVID in that first wave, it's more likely that your sequence is going to be related to a case not found in your extra urban area. It's more likely to be related to other virus found elsewhere in Wales. And then on the end, you can see these are our three genomics indicators, and this information is fed live into Welsh Government, and we've got a sort of a red, amber and green set of criteria which allow us to know what's happening in terms of what the sequence data are showing for Wales, and so that's something which feeds in at the moment. So we're operating across these different levels, and obviously we're interested in what's happening in Wales. So these are case numbers across Wales, across the different health boards, and what we saw in the summer was that like Scotland, COVID was largely eliminated from most of Wales. We weren't seeing very many cases at all. We did see these higher rates in North Wales though, and that was a bit of a conundrum. So we looked at that, and we initially thought that those higher rates would mostly be to do with travel and proximity to places like Liverpool, where you've got where cases were continuing. But actually, what we found was that although sort of 60 to 70% of the cases that were there fell into a small number of lineages. When you started to look at those, we started to identify local outbreak investigations, which revealed other sources of infection. And so this lineage here, the lineage numbers have changed, but the tree is the same. Led us to an outbreak which was happening in a hospital, which then allowed us to actually provide useful information to local colleagues to support the response there. So obviously, after the summer, we had this this lull, and then we saw that the case numbers were starting to increase again, and there was a new spread, and so we've been working quite a bit on how we visualize this. So this is another visualization of different lineages and their frequencies over time. The red blobs are cases in Wales, and the grey around them are cases from England. And what we saw effectively was that the lineages that were present in the first wave were different to the lineages that we were seeing after the summer. And so that raised the question of, well, where did these things come from? How can we look at that? And so, you know, this is something we were looking at over the summer, and it's something that we've continued to refine. And the basic analysis. So what we did was we've used a similar set of approaches. So we've looked for to identify if we can take a set of take these lineages or phylotypes, identify the likely ancestral location of those, and then identify if there's evidence that ancestor is in England or is in Wales. And what was relatively clear was that actually most of those lineages which came through in the summer and now established themselves in Wales were from outside of Wales, and a considerable number of those are from England. We've been doing additional work which has been looking at the sort of time lag. So for the majority of lineages, what we see is we see that there's a time lag where they're observed in England before they're observed in Wales, and that's potentially indicative of them being seeded across the border. And actually, we've been doing a directed analysis to look at the border and look at communities in Wales and comparing that with data from COG-UK, and that's really demonstrated that first that the border is very porous, but also that actually a lot of the flow is from England to Wales rather than Wales to England. And so that's sort of what we would expect based upon the way that travel links work and the way that people move. But it's really important in terms of understanding how the pandemic is being driven in Wales, and also what we can potentially do to try and prevent that. So the broad conclusion from these is that travel over the summer really underpinned reintroductions into Wales, and that like Scotland, we saw the re-establishment of COVID infections in parts of Wales that had predominantly eliminated COVID through the first lockdowns, but also that actually that what happens at the border is really important, and that a lot of the transmission is coming into Wales to establish these lineages, which then cause local outbreaks. And we do in general see a spread from the east to the west, which is consistent with the sort of the transport links within Wales. So we have sort of these two long transport corridors in the south and in the north, and our data suggests that it has spread along those as each of the waves have progressed. So just in conclusion, we've gone from a standing start in March to an operational service for supporting SARS-CoV-2 work in the NHS in Wales. And we did that in quite a short space of time. We've been providing sort of outbreak support and analysis to within both the NHS and to people like Welsh Government since the summer. And that's enabled us to answer all sorts of questions about transmission and what's happening in Wales, to examine local outbreaks and to provide information to a range of different stakeholders. And I think the really important thing is that this didn't come from nowhere. We have a fantastic team. Welsh Government's invested quite heavily in building the genomics capability in Wales. And one of the things that Sally Corden has done brilliantly is to build a platform which is modular, so that when we had to be able to sequence a new pathogen, we could do so and do so with a high level of assurance, because our other clinical services are fully accredited. So it's been an amazing year for us. We've done a huge amount with a very small team, and I'm extremely grateful to their efforts and to the many collaborators who've helped us in our work.
Thank you, Tom. I'll just let that applause die down. Tom, I'm very, very grateful for all the work that you're doing and sharing with us in Welsh Government. It really is impressive, actually. So thank you so much. Look, please keep the comments coming in the sidebar. We've got all these wonderful experts online, so if you've got a burning question, don't hold back. I think there's a prize for the best chaired session, I believe, maybe a small car, and a prize for the best question, which I think was a Kit Kat chunky, I seem to remember. So without further ado, let's invite David Fairley to the stage, and he can share with us his experiences of Northern Ireland.
Good afternoon.
Thank you very much.
Okay, is that on full screen? No. Okay. Well, yeah, good afternoon. Well, first of all, let me also, as with the other speakers this afternoon, begin with the acknowledgements, which I think clearly important that this is upfront and not at the end as an afterthought. First and foremost, I'd like to thank the COG Consortium and particularly Sharon and Ewan and the team they've assembled for the opportunity that it's given us to be part of this. We're an even smaller team than Tom's team in Wales by some margin, I think. But the other people, I've singled a few out here at the risk of offending by omission, but I'd like to in particular thank David Simpson and the small team at Queen's University, who are our core collaborators, and the team in the Regional Virus Lab in the Belfast Trust, in particular James McKenna, who's really pretty much single-handedly kept the flow of samples into our sequencing pipeline moving, and Connell and Tanya in the consultant team, and the Public Health Agency team in Northern Ireland, so particularly Declan Bradley and Tim Wyatt, without whom we couldn't have negotiated the sort of the administrative issues around consortium agreements and so on. Okay, well, conscious of the fact as well that I know that there are going to be many people at this meeting that are new to COG perhaps and haven't seen the slightly bigger picture stuff. I thought it might be useful to take just a slight step back and look at the data as it stood on the 7th of December. So my slides are really based on that data release. So the data releases from COG-UK come thick and fast. In that data release, there were over 131,000 UK genome sequences. The lineage analysis assigns those to UK viral lineages. There's a link on the slide down here, which gives more information for those who'd like to understand how that's done. And then there's another lineage analysis, which is the global lineage assignment. So beginning here, which is assigned using a different method. That's from Andy Rambo's team in Edinburgh primarily, and the publication for that is on the slide also. But just to give an indication of the sort of shape of this, the largest of the UK lineages, which is designated UK 1219, and then or the global lineage B1177, contains 43,000 of the UK genome. So around a third of the UK genomes fall into that single lineage. And then at the other end of the distribution, we have over 1,600 singleton lineages, so lineages that only have one sequence. So a very long tail distribution with a relatively small number of dominant lineages. In Northern Ireland, our sequencing effort began in just around April this year. So since then, I'm very proud to report that we've been able to sequence over 1,900 genomes from a standing start in Northern Ireland. Our lineage diversity is significantly lower than the lineage diversity that we've seen from Matt and from Tom. So we've seen 131 of the UK lineages at least once in Northern Ireland, and 43 lineages 6 or more times in Northern Ireland. So I've gone for plain English, I should emphasize as well, we don't yet have a dedicated analysis team or an analysis function in Northern Ireland, which is something that we're working hard to put in place. Hopefully you can follow this rather text dense slide, but so 11 of those 43 Northern Irish lineages, the lineages where more than 3/4 of the genomes are from Northern Ireland, but 9 of those lineages have been seen fewer than 12 times, so very small numbers. So 6 of the 43 are lineages that we've seen more than 100 times in our data. 2 of them are lineages that we've seen more than 300 times in Northern Ireland, and one of those is the very dominant UK 1219 lineage. 18 of them are lineages that based on our data were recorded either first or only in Northern Ireland, or in GB in the same week as Northern Ireland. So they're immediately interesting as lineages that may be Northern Irish origin. 20 of the 43 lineages remain active in the UK. In other words, they've been seen in the last 4 weeks, and then the smaller group of lineages which may be Northern Ireland associated and which remain active are just 5. So, and I should emphasize as well that in terms of the analysis, that there are lots of caveats around this type of lineage analysis and how much we can infer from it, because certainly in Northern Ireland, our sampling density is lower, sampling heterogeneity. And as we've heard, that there's clear evidence of multiple introductions and underpinned by a relatively slowly evolving virus, which can make this kind of inference very difficult. So graphically, if we have a look at the 43 lineages we've seen in Northern Ireland in any significant numbers, we can clearly see the first wave and the introductions on the left of the slide by date. So those that are marked in blue that's the earliest report of that lineage in any of the UK sequences. Those in green are the earliest report in the Northern Irish sequences, and then the lilac colour is the last reported sequence. So it doesn't mean that these are necessarily extinct, although the sampling across the entire country is clearly very comprehensive. So these are either no longer active or are present in such low numbers that they're not being captured by the sampling and sequencing. And then we can see, as in the other UK countries, a very quiet summer. So the numbers in Northern Ireland, of course, were very low during the summer as well. And then new lineages appearing in our data again as the second wave progressed. Okay, Louis Duplessis, and I have to thank Louis Duplessis in particular in Oxford and also Verity Hill for assistance in interpreting the lineage data. But Louis kindly allowed me to use these slides from these figures from the analysis that he's been involved with. The link to that is on the slide. It's a COG-UK data set, which is available on the COG-UK website. That analysis used a different analysis method to try and assess introductions into the UK. The introductions from Ireland, which are clearly of interest to us in Northern Ireland, given our land border, were part of that analysis and came in using that analysis method in about 6th place. There were a couple of interesting features about that introduction analysis, sort of a step change in the number of potential query introductions from the Republic of Ireland around the time of the first lockdown, although the first figure there is, of course, the proportion of samples. That's not absolute numbers. If we look at the absolute numbers, that appears to have peaked slightly before the UK lockdown. The lockdown in ROI began a week later. But the other caveat here is that the sampling density in the Republic of Ireland is significantly lower again than our own. So the team in the Republic have reported just over 1,000 genomes. So in Northern Ireland, our sequencing cover per capita has been about 8 times higher, and our per capita cover is low in the UK context. So that's going to make definitive understanding of this, I think, potentially difficult. of the Northern Ireland lineages, which are really strongly associated with Northern Ireland, there are two that stand out in our data, both which also map to the transmission lineages in Louis du Plessis and his team's analysis. One of those was a lineage designated UK 601. We've seen 92 of these in Northern Ireland compared to 45 in GB. That hasn't been seen since the middle of May, so is presumed to be extinct or no longer active at least. And a second lineage, UK 1658, which was one of our top two, so the largest number of cases that we had of any one lineage in Northern Ireland fell into this group. Also mapped to one of the transmission lineages in Louis' analysis, but again not seen since July. So that is also a lineage that was very prevalent, extremely prevalent for a period in Northern Ireland, and has subsequently died out. And finally, the last one I've highlighted here is an interesting one, and this is interesting because this is a lineage UK 1030, which, based on the most recent data release, appears to have originated in Northern Ireland. It's now in third place in the UK with close to 4,000 genomes in the UK data set. That also maps to one of Louis' transmission lineages. And the first genome that's assigned to this lineage was a Northern Irish genome from the 1st of April. There was one additional case in England, or one additional sequence from England in the first 10 days of April. It hasn't been seen again. It wasn't seen again until the start of July. And so I think that it's possible that this early placement in the phylogeny is incorrect. And so that this Northern Irish lineage, or if this was introduced initially in Northern Ireland, I don't think it's subsequently responsible for the extensive spread across the border in GB. Okay, so in conclusion, I think we can say that Northern Ireland's lineage diversity is certainly quite a lot lower than the lineage diversity in the other UK countries. We've had mercifully few lineages of note in Northern Ireland, and I think probably the only lineage that looks like it may have originated in Northern Ireland, which has subsequently become very common, probably is not ours. So work to try and understand that lineage transmissions on the island of Ireland, north and south, is ongoing. We're in close contact with the National Virus Reference Lab in Dublin, and so we hope to shed some light on what's happened in Ireland, and with of course our equally porous border in the course of this year as that progresses. So I'll finish there, and thanks for your attention.
Thank you very much, Derek. That was a really, really good, fascinating narrative, and thank you so much for keeping to time as well. So without further ado, I'll invite Professor Deenan Pillay to the lectern to give us a session summary, and then, if we've got time, I'll solicit a few questions from people before we all rush off to join the queue for coffee.
Hi everyone. Good to be here. Thanks for the invitation to join a really interesting and very impressive piece of work and descriptions from the three of you. I mean, it's a shame England are not represented here, and so the theme seems to be keep the English out, to limit spread. But anyway, so I'm Deenan Pillay, it's great to see so many old colleagues and friends on the list here, and I'm also on the governance group that Sharon and Gordon have put together for COG-UK. So I've been following what's been going on with great interest. So I put some slides together as I've been hearing the talks, and hopefully you can see this. A couple of slides with some and apologies for any spelling mistakes, but this is just to stimulate a discussion, of course, and I'll be so. One of the questions we asked ourselves within the oversight committee is just what is the impact that COG-UK can add over and above describing really interesting things, developing fantastic tools to interpret data. But what is the added value on the basis that actually that is the long-term sustainability of the consortium, is demonstrating that value. And there's a couple of things right at the beginning, which is with all the description of the lineages that have been that you've been identifying, to what extent are these just stochastic, introductions that seem to take off in the context of local outbreaks and so forth, which would be expected, versus some sort of selective advantage over one or the other, and what are the conditions which determine that, which of course will be touched on in the characterization sort of sessions later on. And the second is in the context of this huge variability of testing for infections that have happened within the UK, starting with symptomatics, then to some level asymptomatics, as focus on high-risk areas such as healthcare settings, and now this term mass testing used. This leads to a whole range of different data sets which are really difficult to interpret in terms of prevalence. To what extent can phylogenetic analysis really contribute an understanding of that? So I think those are important things, and I was looking at these talks through those lenses, through those lens, that lens. So, and then some specific questions that struck me from the descriptions of the work going on in Scotland, Wales, and Northern Ireland, is first of all what I think is a really impactful observation about the non-UK origins of infections, which demonstrated really the delay, the impact of a delay in border controls early on. Then actually, as prevalence, as overall incidence comes down, vaccines start to have impact. How long should these quarantines last, and to what extent should there be ongoing surveillance of there? But the question is what can COG-UK say to quarantine policy? Secondly, I joked earlier, but in fact you all talked about England, the devolved nation being infected and reinfected from England. To what extent actually, the devolved nation issue has really come to the fore within COVID because of course the separation of health as well as education, and so particularly Scotland, and so that is an interesting question. To what extent should there be ongoing concern of the different policies that are happening across devolved nations? The third thing that Patrick Vallance mentioned earlier, which I've not seen any evidence of, but maybe there is evidence that's coming up here, is to what extent do big movements of people, and universities opening in October and again will be in January, to what extent do those movements really lead to spread then back into the elderly at-risk population? I think that's an important issue. And then impact, clearly there are lots of different non-pharmaceutical interventions, all the tiers and all the lockdowns and circuit breakers that have all happened differentially across the devolved nations. To what extent can we say that COG-UK can actually speak to how effective those have been in terms of impacting on lineage growth? And then the final point I'll make, and in relation to, of course, the recent description of a growth of a lineage in London and the southeast, is how able is COG-UK really to be able to distinguish between a lineage that grows because of a viral characteristic versus a lineage that grows because of epidemiological growth and spread because of contact between people. So those are questions. And then the final thing in my next slide, if I can move, is just then, of course, the critical thing in making sense of all this other metadata. And I'm well aware that it's not straightforward to have access to metadata, but clearly there is a need for issues around detailed prevalence and from testing data. Mobility, since much of the data that you've all talked about have been around identifying routes of mobility determinants of that. In Wales, I was struck by that transmission along the routes, the traffic routes. But of course, with Google data, there's a lot of activity in this area. To what extent can there be a link between lineage growth and actual understanding about mobility of people? And of course, the critical thing is location and other associated demographic data associated with each of the samples that come. So these are just questions of to what extent do you have access to these data, and to what extent is this required in order to develop the sort of the impact sort of COG-UK, which I'm sure there's huge potential. So hopefully I'll stop there, and hopefully that's precipitated something of a discussion. Thanks a lot.
Thank you, Deenan. That was really helpful, and so good to be able to add that contextual narrative of, you know, where we need to go next, and what we've learned as we've gone along. So hopefully that's stimulated some questions. If not, I will absolutely hog the stage and ask lots of questions. So if there's any hands up, anybody wants to come in with a question, please speak now. Forever hold your peace. I'm hoping people are still with us. It's difficult and you can't see the faces, knowing whether people are watching Christmas Vacation at the same time as listening. So any hands up?
We have a question from Sharon, Rob.
Okay, Sharon, please feel free.
Thanks, Rob. I just wonder, I mean, the great thing about having the MRC Climb database is that data from all around the UK get put into the same place, and that's a real kind of a bonus of what we're doing. But I wonder how readily data from different nations actually flow into each other, and whether there's anything we need to be doing there to improve that. Because I think my understanding is that actually each nation has its own kind of data system. And I think that Deenan's point about data is absolutely critical. You can only really make sense of the genomes once you've got some metadata associated with it. And so, although it's not a genomic question, data is so central to what we do. I'd just be really interested in people's observations about whether we could be doing better with that.
Thank you, Sharon. Who wants to come in? Maybe you've stumped them, Sharon. Thank you. So if anybody has any reflections on Sharon's question, please feel free to pop it in the sidebar. I wonder, Sharon, if there's more that we could be doing through the HDR UK structure at a UK level to bring more data sets together.
Yep, absolutely.
I will just jump in with a reflection. I mean, obviously I'm conflicted because I'm obviously part of Climb as well. But I mean, I think that the thing that's really been sort of transformational with what we've been doing is that, you know, obviously there's 4 separate kind of healthcare systems in the UK, and you know that I think there's layers of data, right? So HDR UK hold very sensitive data, but it's very restrictive what you can do with it. Right, so you know it's hard to, it would be hard to ask some of the sorts of questions that we are interested in HDR UK, because it's much more, like, these are much more speculative. And what Climb has let us do is to sort of collate together less sensitive data, which still requires some protection and some processes in place, but then facilitates much more fulsome analysis in response to need without needing to go through a whole process to get those studies approved. And so so Climb sort of is in this sort of like this position between, you know, being fully open where you can't release very much information and holding some data which could potentially be sensitive, but which can be stored in Climb in a more limited way. And I think we've really, you know, that this problem of how we share pathogen genomic data more generally, how we analyze that sort of collectively, sort of across borders, has been a problem from a sort of an epidemiological and more sort of broader global health perspective for some time. And actually the solution that we've got with Climb is really interesting, because it does create real opportunities potentially for building on that and improving the ability to share genomic data and support more global epidemiology type work.
Thank you, Tom. Absolutely unbiased opinion there of Climb, but thank you very much. I'm going to take one last question, then we're going to break. So, Professor Carol Mundell, the floor is yours.
Thank you. I'm Chief Scientific Advisor at the Foreign and Commonwealth Office, and it's a great pleasure to be here. Thanks for the invitation. What I really wanted to just reflect on was the incredible power of the genomics data so far for giving us insight into the nature of our own pandemic. And I know there'll be discussions about universities and international borders. I'm not a virologist by background, I'm a physicist by background. I think of this virus in a very physical way. And what has really struck me is the ongoing lack of understanding that many people, including academic colleagues who I don't consider to be unintelligent, they just don't understand how the virus, how it spreads, how it's transmitted. And I think I also see sort of many misnomers in many of the groups that I interact with in terms of what works, for example, MPIs, what the nature of our epidemic was. And I think, the genomics data for me right from the beginning, particularly when you published your first papers showing the introductions into the UK and the impact that they had, I think they also really changed the way our epidemiologists thought about this infection. I know early on in SAGE, there was less focus on introductions. It wasn't thought to be that critically important, I guess, until after, once you have community transmission, then that takes off and that's where you focus. And I think it was really that insight that came from the genomics work that helped us to think very differently about that. So for me, I always think data are not information, information is not knowledge, and knowledge is not insight. And it's really insight that we're trying to get to. So I'd certainly second what Sharon says about, the data and the data flows. And then also thinking, I think from your perspective and how we can help you in government to connect through to the operational side, to make sure that those insights are accurate, but those insights also really impact on the way we design our systems. This is the first time we've ever had this kind of pandemic in our country. I think that shows we're stepping up these systems literally from scratch at pace, which for me has been incredibly impressive. But it's also, I'm also mindful that one can either lose that or it becomes an academic exercise and lots of academic papers get written. How do we make sure that that stays at the forefront of sort of our operational response? And I know the likes of Rob and Sharon, we're sort of all closely involved within SAGE. And I think making sure we keep having those conversations is really important as we go on. So just thanks to everybody for the work you're doing. I'm learning a lot. I'm reading lots and lots of papers. And obviously there's then the question that we move into sort of pathologicity and what that actually means for people's health, in addition to just actually understanding how things spread at that forensic level. And also a big shout out to everybody who's helped on animal-human interface, particularly around Danish mink. As a vegetarian physicist, I never really thought I'd be having to learn so much about deskinning and removal of bodies and shuddery things like that. But again, the genomics sitting really very much at the heart of that. And I think the UK is a big world leader in this, and we're trying to make sure that other countries start to step up their systems, both at the human side and also the human-animal interface. That will be a long-term legacy from this pandemic. Thank you.
Thank you, Carol. Carol, it's lovely closing remarks there as well. I'd really like to take this opportunity to thank Sharon and everybody in COG-UK. It's absolutely incredible piece of work. So just a huge thank you. I believe we've got a sufficient time to have a 6 minutes. That's right, 6 minutes break before the next session. So make yourself comfortable for, I'm sure, a very enjoyable session led by David Crossman after the brief break. So please put your hands together and thank all our speakers this afternoon, and see you back at in 6 minutes. Thank you. Bye.
Hello, good afternoon. My name's David Crossman. I'm very honoured to have been asked to chair this second session, which is about transmission in special environments. And there was a nice segue into that from Deenan's summary. I'm going to allow myself just a couple of minutes to say a few things. First is I want to thank Sharon, really on behalf of probably everybody, but certainly on behalf of the devolved administrations. It's the UK in COG-UK that's really important. And I must say, Sharon, thank you very much for exemplary involvement of the devolved administrations, which doesn't happen all the time, let me put it that way, and it's much appreciated. And I noticed that you've got the chief scientific advisors from each chairing a session just to reinforce that. The second thing is to reflect, I probably can say this, is that I can remember sitting at the National Genomics Board where we had to discuss whether infectious agent sequencing was really mainstream in a national genomic strategy. And I said to Mark Caulfield back in April, I think it was, I said, well, we're never going to ask that question ever again, are we? When COVID came along. And amongst the many things COG-UK has done, it's absolutely proof positive now that we can't manage our science of infectious disease without this type of approach. So powerful was it, I was reminding myself when Matt Holden was talking that I managed to get Matt to come and present to the First Minister. And Matt's got a certain way of presenting, but sometimes complexity is a feature of his talks, and he didn't spare the First Minister the complexity, I'm pleased to say. Anyway, such an approach allowed funding of a service within the NHS within Scotland. And so she was persuaded. And I think the final reflection I want to make is this pandemic has really held up to us. If we really want to do something, we really can, and we can be really world leading. We've been world leading in this, we've been world leading in the recovery platform for new therapeutics, we've been world leading in vaccine development, and we've been world leading in human genome sequencing with Kenny Bailey's paper in Nature last week. We don't want to lose this moving forward, and you know, with this power and effort, I really do believe that egregious issues such as dementia and things really could fall away. But I think that's for us to press our scientific leaders when we've got over this current pandemic. But I think it's an incredible effort from everyone. And I think the UK, amongst all the things that are going on, can really hold its head up high. And it's people giving these talks and the team at COG-UK and Sharon's leadership that has certainly enabled me to be able to say that. So enough of that, but those words were sincerely meant. And we're going to move on to the special environments. And Estee Torok is going to tell us about hospitals. Over to you, Estee. Thanks.
Okay, thank you very much indeed. I'll just share my screen. Great. Okay, well, thank you very much to Sharon and COG-UK for inviting me to give this talk. So I'm going to describe some of the work that we've been doing in Cambridge since the start of the pandemic, particularly using rapid sequencing to investigate healthcare associated infections in Cambridge. So this is all very familiar to people. Obviously, the virus emerged a year ago and has infected over 70 million people worldwide and caused over 1.6 million deaths. The burden and impact of healthcare-associated COVID-19 infections is unknown, but is estimated to represent up to 18% of cases, with a very wide confidence interval. In Cambridge, we aimed to examine the utility of setting up a rapid SARS-CoV-2 sequencing service and to combine sequence data with detailed epidemiological analysis to investigate healthcare-associated infections in our hospital. So how did we do this? Well, we did this with an Ian Goodfellow's lab at the University of Cambridge, and we set up a rapid SARS-CoV-2 nanopore sequencing pipeline, which was sort of modelled on the work that he'd done in West Africa during the Ebola epidemic. And what this enabled us to do was it enabled us to go from a PCR positive diagnostic sample, which we obtained from the Public Health England microbiology lab, which was located one floor above Ian's lab, and brought them down to Ian's lab, where we were able to amplify and then sequence these samples so that we could go from PCR positive sample to sequence in under 24 hours. So we obtained samples from the PHE lab initially. As we set up a healthcare worker screening service, we then included the healthcare worker screening samples within this workflow. And then finally, Ravi Gupta developed a Samba point of care test, and we also aim to include those samples. More recently, we've moved over also to using discards from the Panther Logic platform as well. So what happens is, in terms of the sequencing side of things, it takes sort of 1 to 2 days for us to go from sample to sequence data. And then we used to analyze the data and aim to provide genomic and epidemiological information within 7 days or so. And the work that I'm going to present was the genomes that we analysed up until the end of the 24th of April, so about a month and or so after we started sequencing, and that included 299 genomes from Cambridge University hospitals and 46 healthcare workers. Oops, it's gone the wrong way. So this slide basically summarises what we did. So up until the end of April, we'd done about 24,000 PCR tests, of which about 5,600 were positive, including 374 inpatients. Of those, we managed to sequence about 1,000 samples and generated 747 SARS-CoV-2 genomes, as these were the ones that sort of passed QC and had metadata and were not duplicates. Among these 747 genomes, there were 299 samples from patients, of whom 37 were healthcare workers, and we also included 448 samples from the east of England. And to provide context, in terms of the analysis that we did, we compared the inpatient genomes and the healthcare worker genomes in order to look at evidence of hospital-acquired infection. So this slide shows the epidemic curve of COVID-19 at Cambridge University hospitals. And what you can see is a sort of classical epidemiological curve shape, with the number of cases rising and then falling over time. I'm sure you're well aware that we went into lockdown on the 23rd of March, and the number of cases continued to increase before it then declined as lockdown measures took effect. The other thing that you can see in this slide is that initially our samples were dominated by community-acquired infections, but the number of healthcare-associated infections increased over time, and we also had a fair number of infections among healthcare workers. The legend on the right basically shows the different categories of healthcare-associated infection, which are shown in colours, the colours other than green and pink. So first of all, I'd like to show you a phylogenetic tree, which demonstrates the geographical structure of the viruses that we examined. And what you can see is that, first of all, if you look at on the left-hand side of the panel, you can see that there are certain clusters of infection that seem to be related to specific wards, so as indicated in the colours, so wards A, C, and the dialysis unit. And when we examine the pairwise SNP differences, you could see that they were very closely related, with a maximum of 0 to 1 SNP differences between them on wards A, C, and the dialysis unit. And this compared with a wider range among samples taken from the whole of east of England, from Cambridge University hospitals as a whole and from the emergency department. And our view is that identical genomes are unlikely to occur by chance. So the chance of any two randomly selected genomes from this study being closely related was estimated to be 4.5%. And what we did then was to look in more detail at the clusters. So this one, first of all, I'm going to tell you about a cluster that happened on a ward that we designated as Ward A. This slide shows an epidemiological timeline of the 6 patients involved in the suspected outbreak. So what you can see is on the 27th of March, patient A becomes positive for SARS-CoV-2, and then a couple of days later, patients B and C also test positive. A few days later, patients D, E, and F also test positive, and they are all located on the same ward. So this, to anybody, is pretty much pretty likely that it's a ward-based outbreak. And the interesting thing about this particular ward was that all of these patients had been in hospital for at least 7 days, and some of them for much, much longer, suggesting that this was very much hospital-acquired infection. And the genomics basically confirm that. The second outbreak I'd like to explain is the dialysis unit cluster. So this is, again, we found a number of genomes that were associated with a renal dialysis unit. And the thing that was slightly unusual about this was that the patients all presented within the same 2-week period to the emergency department and to various wards. But in fact, when we looked through the epidemiological data, even though they were diagnosed either in the ED or on the renal ward, we discovered that all of them had dialyzed on the same outpatient dialysis unit on the same day. And a detailed epidemiological investigation, in fact, discovered that they had all been transported to hospital in the same transport vehicle, and obviously they had to sit in a waiting room as well. So a number of infection control interventions were instituted, including universal masks for staff and patients, closure of the waiting room, improved social distancing, and improved transportation. And to date, we have had no further cases on the dialysis unit. And finally, I'd like to mention the power of genomics in terms of investigating community clusters. So here you can see highlighted in the phylogenetic tree 18 cases that had identical genomes. When we looked at the epidemiology, 9 of these were resident in a care home designated Care Home A. 1 individual was a care home worker. Another one was a care home worker in an unspecified care home. 3 individuals were paramedics. 2 were healthcare workers who worked at Addenbrooke's who lived with the paramedics, and 2 were spouses of cases, but with no identifiable contact with others in this cluster. So this basically highlighted the power of genomics to identify a genomic cluster, which when we looked into in more detail, we found epidemiological links between most of the cases. So in terms of the summary of our genomic findings at CUH, we identified 35 clusters of genomically identical SARS-CoV-2 samples, and this represented 53% of our sequences. We identified transmission events on 12 wards, 3 of which were red wards and 9 of which were green wards. I've obviously only given you a couple of examples of those. And we also identified transmission on a dialysis unit. I've also shown evidence of community transmission network, which involved a care home, family and household contacts. And also we highlight that healthcare workers were involved in both community and hospital transmission events. So I just want to talk a little bit about the investigation process at Addenbrooke's. So all patients who are admitted with confirmed SARS-CoV-2, we basically look at them and decide whether they could have possible healthcare associated COVID according to the PHE definitions. The cases are then reported through the standard hospital reporting system, and one of the infectious diseases registrars goes through the notes to see if there are any reasons why the healthcare associated infection may have happened. In parallel, the Public Health England Field Epidemiology Unit did an epidemiological analysis using their cluster track software to look at patient contact on wards and to determine whether there had been contact in certain locations. And in parallel, we conducted a genomic analysis of any samples for which we had genomes. And we would basically meet once a week on a Tuesday to decide which patients we would look at and which wards we would look at. And then we would present our analyses to a hospital meeting on a Thursday. And this involved infection control, clinical doctors, the patient safety team, and the genomics and the epidemiologists. And this enabled us to discuss the healthcare associated infections, and also to see if the genomic data supported or didn't support evidence for healthcare associated infection. It also enabled us to further target investigations and also to target infection control management according to the findings of these meetings. So just to summarise, we set up a real-time genomic surveillance programme for SARS-CoV-2 at Addenbrooke's Hospital. This identified transmission networks which involve both patients and staff in the hospital and in the community. And we integrated this with epidemiological and clinical data of individual cases into a patient safety review process and identified opportunities to target infection control interventions. And I'd like to just finish by thanking the many, many people who were involved in this programme at Addenbrooke's, in particular Ian Goodfellow and his laboratory, who did all the sequencing, a number of people within the Department of Medicine, the PHE Clinical Microbiology Laboratory Lab, and also the university and the field epidemiology unit. Okay, thanks very much.
Estee, thank you very much indeed. We will do any questions or things at the end, so we'll move on, but I would just like to reflect, this shows that you can do things about apparently egregious issues, and I just think the dialysis unit story is brilliant. Really sick patients, really high risk, seemingly very difficult problem, simple MPIs make the problem go away once you know how to deal with it and measure it. So congratulations to you. We'll move on. We'll go a little further up the road to Norwich from Cambridge. And Andrew Page is going to present data on care homes from the Quadram Institute. I remember the Quadram Institute being built when I was in Norwich, so it's rather nice for me to hear that. So over to you, Andrew.
Thank you very much. Hopefully you can see my screen there and hear me. So I was asked to undertake a care home review with a colleague from the University of Cambridge, Dinesh Agarwal. And what we did was we looked at all available studies that used genomics to look at care homes. And of course, care homes is a big problem because there's a huge amount of mortality associated with care home residents. So the first thing to note is that in the entire world, there's only 11 studies and only 3 countries represented. So there's huge gaps in this field, even though it accounts for such a very high proportion of mortality. So much is to be done. The UK, of course, has done the most genomics, so we're the world leader, I suppose, in this. Mostly people are focusing on small outbreaks as well. So I just want to go through a few examples, of where it's been useful and kind of things you can do with genomics when looking at care homes. So the first is a study from Washington. And what they did was they went and tested everyone and then put all the positives, sequenced everyone, put all the positives onto a tree. And then they just simply labeled all of the sequences and then overlaid those labels on top of a map of the unit of the care home. And what it identified was that actually residents whose bedrooms were quite close together were more likely to be infected with an identical strain, which makes sense. But when you consider that there were extreme infection control measures in place, like residents had to wear masks when leaving their bedrooms, and staff weren't moved between units, and then anyone who's positive was moved elsewhere, it became apparent that it's quite difficult to control. This, of course, was very early on. So genomics at a very simple level can be very, very powerful. A study then from the east of England, from Estee's group actually, looked at care homes, except on an insane scale. So this is by far the largest study by many orders of magnitude, and they looked at nearly 300 care homes and found that there was actually more than 400 lineages in those care homes. And there were a lot of different independent introductions. And when they went and looked back, they found about 6% of the infections were suspected hospital-acquired infections. So possibly a bit lower than the media and dogma would have you believe. So quite an interesting finding. And of course, they did find some evidence of some inter-care home transmission, as she previously mentioned. In Norfolk, we did some work on a care home specific lineage, and it came about by accident. We were asked to look at one care home outbreak, and then we scratched the surface and we found 6. And all of these care home outbreaks were linked, all the samples were either between 0 and 3 or 4 SNPs apart from each other. So they're very, very similar. And we had this like 100% identical genomes in multiple different care homes. And on the right-hand side, you can see there's like colors to represent different care homes in different categories. So that's a bit of a problem. And the way we found this was doing some very simple analysis. We just looked at a heat map of the postcode data and found some very elderly people, very old people living in particular postcodes, and figured there might be a problem there. And then digging into it and doing a bit of discharge analysis with Public Health England, we identified actually there are only 2 probable hospital-acquired infections and 1 suspected. So that gives us an indication that that hospital discharges played a small role, but mostly it was most likely inter-care home transmission. We don't know what route that took. It may have been shared staff or shared professional visitors. But luckily this care home outbreak is now extinct. We did a bit of analysis on actually just some methods people were using in all of these different studies. And there's a huge variety. One thing that does stand out is that in a few cases, there was errors in the way that the bioinformatics and sequencing was performed. And that's mostly down to maybe people coming in from other fields, not realizing that you need to do things slightly differently when you're dealing with viruses. And that is a problem, and it's something we all need to work on as a community, because erroneous results will obviously give totally wrong data at the end. Another key thing to pick up from this is that a lot of studies on coronavirus are simply not reproducible. Either the data is not available, so people haven't made it publicly available in the standard archives like GISAID or the ENA, or they've provided incomplete metadata, so you can't actually link up the samples that are publicly available with the samples described in papers. And that prevents reanalysis, maybe on a larger scale. And that is something we really do need to work on. COG-UK, luckily, is doing quite well on this because we release all of the data as we go along. So the overall key findings for this review, there are many, many findings and recommendations in our paper, but the kind of headline ones that you should take away from this is that there were about 6% of infected residents were suspected or confirmed hospital-acquired infections. We found usually staff and residents had the same lineage and usually identical genomes when there's an outbreak in a care home. In fact, even staff with no patient contact usually had the same genomes. So that is a problem. So PPE and infection control needs to encompass everyone, not just staff who are caring directly for residents. There was a lot of there's limited genomic diversity. So usually you would get, one lineage or would dominate, and you might get 1 or 2 others sneaking in, but they wouldn't go anywhere, rather than kind of a continuous flow inwards. There were some shared clusters between care homes, and that was identified in multiple studies. So, you don't want to share staff, for example, between care homes, and you want to limit that. I know in the UK, they're trying to prevent that. And also with some serial studies, they found that even when it appeared like they got rid of all the cases and there's no more positives in the care home, the same lineages would keep popping up again and again. Now, it's unclear whether that would be because it was just circulating in the community and it kept coming in, or if it was hiding out somewhere, or if people were just being missed who were positive. Overall, the mortality rate was high, and it didn't matter what lineage was in a care home. So it's unfortunately just the comorbidities were the problem. Bioinformatics methods were misapplied in up to 1/3 of studies, and we need to work on that. And we can't reproduce a lot of what is published. So this review was the work of quite a lot of people, in particular Dinesh in Cambridge, and we had a lot of help from the PhE as well. So thank you very much.
Thank you very much indeed. And thank you, Andrew, for raising the, I don't know if it's the spectre, but the wicked issue of standards and reproducibility. And maybe Gordon will pick that up in his summary session. I'll leave that hanging there, but always an issue in new technologies, whether that's this type of analysis, bioinformatics, or just new technologies. But we'll move on with time, and Ben Warne is going to tell us about universities. University of Cambridge led the way by a very cunning bit of negotiation with the Lighthouse process, and is the envy of pretty much all other HEIs in the country. So Ben, really interested to hear what you're going to say. Over to you.
Thank you very much. Thank you for the opportunity to present this. So we have been quite extensively studying our university population as part of our COVID-19 measures over the last term, and I'm going to present to you part of this collaboration between COG-UK and the university to show you what we've found. We're the first of a number of universities and higher education institutes across the UK who are working with COG-UK for genomic investigations. But I think that we are slightly further advanced, and I'm delighted to be able to present our findings with you today. So universities are interesting. They provide a number of challenges in understanding transmission events among this population. So you have a population of young people who have travelled from many different national and international locations into the same spot, where they are often housed in very dense accommodation with high risks of transmission, and are more likely to participate in activities that are also high risk. So things that we know are high risk, like contact sports, possibly poor compliance with social distancing measures, etc. We also know that there's very likely to be a substantial role of asymptomatic or poor size symptomatic transmission between these individuals, and their population are themselves unlikely to develop severe disease. So they are often a group of people that perhaps where case ascertainment is very, very difficult to be able to accurately determine. Where genomics can step in is hopefully be able to help to understand how the virus is introduced into these university populations, whether and how there is transmission between the university and the surrounding community, and how the virus is transmitted within the university setting, in accommodation, courses, and other settings that are common among university students. So in Cambridge over the last term, we have provided two pathways for SARS-CoV-2 testing. The first is a symptomatic pathway where any student or staff member as part of the university could get a test free of charge from the university. One of the key things about Cambridge is that all of our NHS testing facilities are outside the centre of the city, which means that the majority of our students would use our own facilities as their first point of call. And then secondly, we've developed an asymptomatic pathway led by Nick Matheson, where we've been aiming to screen as many individuals as possible for COVID-19 to try and combat this problem of asymptomatic transmission. And I'm going to go into a little bit more detail because it's a little bit more complicated. So in broad overview, we aimed to screen all of our undergraduate and postgraduate students living in university or college accommodation. So the university is divided into 31 colleges. Each student has a college affiliation where they live and socialise. as well as having separate departments in which they undertake their university courses. And roughly 2/3 of our students live within college accommodation. They are broken up into households where the average household size is 7 students. And we aimed to screen each household on a weekly basis during term time. And we started off by screening on average 2 students per household. And by the end of term, we were screening all consenting individuals within that household using self-administered nose and throat swabs, which are taken within the student's own household. Now, a key feature of this program is that to maximize our testing capacity that we've had in agreement with the local Lighthouse laboratory, we pooled our samples at the time that the samples were taken based on household structure. So in a household of 7, we would, for example, have a pool size of 7 students where each student contributed a single swab into the same test tube. We would then test that tube. If it was negative, we would just continue to get them to repeat the following week. And if the test was positive, the pool test was positive, we then went on the following day to test each individual within that household or testing pool to identify the individual household member, identify the positive case, and then use that to guide isolation and infection control measures. And it was those individual samples that we went on to use for sequencing. So to break this down into numbers, we have roughly 23,000 students in the university and 12,000 staff. Of those 23,000 students, roughly 2/3 live in college accommodation, and over 80% of them by the end of term had consented to participate in our screening program. And we estimate that we've screened each of those consenting students on average just over 4 times over the course of the term. And the graph on the right just shows the number of eligible students in red, the number of consenting students in blue, and the number of students that we screened each week in black. So by the end of the term, we were screening over 9,000 students per week. We identified 260 asymptomatic students through this pathway, in addition to over 500 students as part of our symptomatic pathway and 28 staff. So in combination, 791 confirmed positive cases, of which we have complete sequence data for nearly half of those so far. We're still mopping up the end of term at the moment. So the sequencing pipeline may look a bit familiar because it's the same as the one that was used for the healthcare worker screening program that Dr. Torok mentioned earlier. So all of our isolates were extracted at the local Lighthouse laboratory. All of our symptomatic, asymptomatic pooled individual tests. The extracted RNA was then taken to Ian Goodfellow's lab in collaboration with Sharon Peacock's lab in the University of Cambridge, where the isolates were sequenced using nanopore and then analysed by teams here in the university. They were then placed in the context of healthcare worker and hospital isolates that was sequenced using the same pathway that Dr. Torok mentioned earlier. And we also used other contextual NHS testing samples that were processed at the Wellcome Sanger Institute contemporaneously with the isolates that we were identifying at the university. This is the epidemic curve that we had over the course of the term. So the symptomatic cases in red and the asymptomatic cases in blue, these are total numbers. And you can see that we had 2 spikes in cases. The first was the second week of term, so the week after Freshers' Week. The second spike occurred in the week following the lockdown, where there were a number of incidents that we think may have contributed to both the spread of the virus within the university. Of the 791 confirmed cases, the majority were undergraduate. There were a number of postgraduates and a small number of staff. There may be some ascertainment bias there, but we think that that probably does reflect that the largest burden of cases was in undergraduates. And of those undergraduates, roughly half of both the symptomatic and asymptomatic cases were in first-year students. So to date, we've analysed data from weeks 2 to 5 of term. So this is essentially October and the very start of November. And this is the phylogenetic tree that we've produced to date. Each node is coloured by the college in which the student lives or is associated with the green nodes reflecting contextual isolates from the local community and the local hospital. So a few things to notice. We have 219 genomes in this analysis. And I've identified as part of this 12 clusters, which include more than 2 individuals, and 4 clusters with more than 5 cases. By the second week of term, the vast majority of our university isolates were confined to only 3 lineages, suggesting there's very limited seeding from students returning to the university at the start of term. And to date, we've identified very limited transmission between students in the community. So of those 282 contextual isolates from the community, only 15 cluster with university isolates. And we are currently working hard with colleagues in Public Health England to try and ascertain whether those 15 individuals had associations with the university which have not yet determined. But certainly there's no evidence of sustained transmission between the university and the community identified up to the 5th week of term. In terms of transmission within colleges, we've identified 2 large outbreaks associated with colleges. So this is the first in College A, where we've identified 37 confirmed cases in 1 accommodation block within 1 college. That was part of the asymptomatic screening program where the first cases were identified. And then we performed a targeted screening program in collaboration with Public Health England to try and identify as many cases as we could in this accommodation block. And 31 of those cases belong to the same genomic cluster. The second cluster in a second college, where we had 22 cases, 10 of which were from the same college. And the majority of the remaining 12 cases could be linked to students from College B due to sharing the same course or the same relationship, and other sort of close epidemiological links. And interestingly, 1 household from College A was also was related to this cluster rather than cluster 2 as I mentioned before. What's very reassuring is that the interventions that were put in place by the university to try and prevent spread beyond the colleges seem to be ineffective, because by the end of week 3, we had no, the lineages have become extinct. There's no further evidence of transmission of these 2 lineages. However, there's this very large cluster of cases right at the top of the tree here, which represents quite a significant sort of transmission events between colleges. So zooming in a bit further, what we see is in week 2, highlighted here by this red box, we had 29 cases, and even by week 2, they had spanned 14 colleges, 23 households, and over 13 courses. And by the end of week 5, they made up nearly 2/3 of all of the cases identified up to this point, and it was essentially endemic within the university. We're currently working very hard to try and determine why this was already established by week 2 and whether there were any events at the very start of term which might have precipitated this transmission. But certainly this has now become the dominant lineage within the university. And there are some limitations to the analysis we've done so far. So there are obviously issues regarding sampling, and there is an attrition of samples through quality control screening, and a small number of students may attend NHS testing facilities. And today we haven't been able to identify any strong conclusions regarding causal relationships between colleges, courses, and cluster propagation. We are currently analysing weeks 6 to 9 and contextual samples up until mid-December to investigate further this issue of community and university transmission. And we are trying hard to identify further epidemiological associations through as many different routes as we possibly can. And there are questions over whether this data in Cambridge is generalisable to other higher education settings. But I think there are certainly lessons that we could be learned from Cambridge that may easily transfer anywhere else. So in conclusion, to date, we have identified very limited transmission between students and the community. Diversity among students is low, so there appear to be very few introductions that led to established transmission. And through successful interventions, we've managed to cause cessation of outbreaks, which have been largely restricted to a single college. But this large cluster, which appears to have affected multiple colleges very rapidly, is much more difficult and appears to be the ongoing source of transmission by week 5. And as I mentioned at the start, this is one of many projects that's ongoing in a number of UK universities. I'd like to finish off by thanking a very large number of people that have been involved, both in COG-UK and across the University of Cambridge. It's been an enormous effort on many people's parts over the course of the last term. Thank you.
Thank you very much, Ben. Fascinating. This would be a very good test bed to work out why a dominant strain becomes dominant. The question that Deenan asked in his summary of the previous session as to whether this is just stochastic or whether there's some selection pressure, I think that's very interesting. I was musing the distribution between undergraduates, postgraduate and staff and risk taking. And as an observer of somebody who's never worked in Cambridge, the risk-taking behaviour of students not wearing cycling helmets is one that leaps out at you. And I wonder whether you could do a correlation between cycle helmet wearing and transmissibility. But that's a bit tongue-in-cheek. Thanks very much, Ben. We're going to move on to Sentinel surveillance using Pillar 2 samples. And this is from Sanger, and Jeff Barrett is going to take us through this. Thanks very much. Jeff, are you there?
Yeah, I'm here. Thanks.
Very
Can you
good.
see my screen?
Wonderful. And I can see the screen. Thanks.
Great. Yes, I'm going to talk about Sentinel surveillance work, which is Sanger's primary contribution to COG-UK. And the reason that we undertook this was broadly in two groups. One is we think there's some evidence that the genome sequences, if produced fast enough, can actually make a contribution to the kind of live detection of outbreaks. That's a hypothesis to be tested. And the other is to essentially monitor for the development of interesting mutations. So to do that, one principally needs a large random selection of positive tests from around the country. And as has already been alluded to, there's at least one example of such an interesting mutation that this surveillance contributed to. And I'll just mention that URL down the bottom. There are over 200 members of staff at the Sanger Institute who have made a contribution of one kind or another to this. So it's really an amazing team effort that's come together in the past 9 months. The UK is clearly the world leader in this kind of use of genomic surveillance, and this sort of pipeline that we at Sanger, together with the Lighthouse Labs, have built is driving that. And it is really a critical thing that there is this Lighthouse Lab network of high capacity centralized community testing. And what Sanger set up early on was daily couriers to essentially take waste material from the PCR tests, bring them to Sanger. The second piece is having a large capacity genomics facility. Sanger first has to pick the positives from all of the tests. So that's sort of step one of the processing, because we get just the general test residue. Then secondly, that has to be, we have to sort of identify the subset that we're actually going to sequence, and I'll briefly mention what our current protocol for doing that is. And then the third step is to generate the genome sequences, and we both have a high throughput pipeline at Sanger, and we also distribute these Pillar 2 samples to other COG-UK sequencing sites who have capacity week by week, so that we can kind of try to get as many of these done as possible. And you can see some of the numbers down the bottom of our current run rate. We get about 500,000 samples per week arriving on campus. We can pick about 20,000 positives out of that. So that's at the moment clearly not all of them. And then we can get sequences about 6,500 or between 1,000 and 2,000 on a typical week at Sanger and the rest of COG respectively. And these are constantly being uploaded to the Climb server so that everyone in the consortium, and indeed everyone in the world, can access these data. And just to give you some sense, these vans arrive. We have these large freezers, -20 freezers we installed on campus back in April or May. These samples arrive in giant green crates, and they are full of 96 well plates that have come straight off the back end of the testing pipeline. And essentially the way we're doing this is at the moment taking a kind of random mix of crates from each of 4 of the Lighthouse Labs in Alderley Park, Milton Keynes, Cambridge and Glasgow each day. And we take all the plates out of the crates that we've chosen, and we pick all the positives off them and sequence as many as we can. So it's not, totally designed as a random algorithm, but it is a quasi-random selection of all of the Pillar 2 samples. And I think it does a reasonable job week on week of getting good coverage around the country. So this is then the picking from those plates to get them into consolidated sets of just positives. And if you look at the volume of sequencing that's come out, we kind of really started this in earnest at the beginning of August. And we've kind of week on week been increasing to the point where in the first half of November, we were at Sanger alone running well above 6,000 a week. These are the dates along here are the test dates. So of course the data kind of flows on a lag of about 2 weeks behind that. The big dip you can see at the end actually has 2 explanations. One is there's just a little bit of data lag, so that will go up as we kind of add more sequences. But the other, as those of you who run sequencing facilities will know, is there is a global shortage of disposable pipette filter tips, and we go through these at an astonishing rate. And in fact, we've had to reduce our weekly volume for the last couple of weeks because we simply don't have enough tips to keep sequencing. The other aspect of this is how long does it take? And this does have a slight downward trend, although it's not that steep. But basically, from the moment the swab is in someone's nose to when the data is on climb and can be used is what we think is the key time window. We at Sanger obviously don't control all of the factors in this, but it's the sort of biologically most relevant time. And it has been going down from about more than 2 weeks early on to now about 11 or 12 days on average. And we hope that we can continue to drive that down, because obviously getting things done within a week would be a really big and valuable improvement. And to that end, we're in fact moving our operation to a new facility that will open in January. On the left-hand side here, there's a number of these new Beckman robots, which will increase our cherry picking and our cherry picking capacity and hopefully our speed. And this lab is going to be operational in January. I don't know if Nick Lemon's on the call, but I took this picture because this crate appears to be absolutely full of tips, and he'll probably be envious. So what does this produce, as well as just putting the data into the hands of everyone else? We've been building some reports that essentially look in a rolling 7-day window at the diversity of sequences around the UK. And we calculate a pretty simple statistic, which I won't go into in detail, but happy to take up offline, which is if you look at any given postal district or area in any given week, you have a bag of sequences, and you can ask the question, if you draw randomly from that bag, how likely are you to see the same thing twice? How homozygous essentially is the sequence mixture that we've sampled in that place at that time? And we use an empirical distribution of what's happening everywhere in the country to simply ask how far into the tail of the distribution does this sit? And we can therefore color different regions yellow if they're in the kind of high end, and red if they're in the extremely high end. And we can look at this map week by week. And this particular set of visualizations comes from a postdoc, Matthew Sinnott, in the group. And then you can sort of zoom in. This is data up until sort of mid to late November. If you zoom in any one of these regions, you can see a bit more info. So here's Medway, which is in the southeast in Kent. And you can sort of see over time, this statistic of homozygosity has gone up. It was very high starting in late October, and it's kind of bounced around, but is still at present extremely high. And what this means simply is that if you take random samples of sequences from that region, you will see the same viral lineage over and over again. And you can get a bit more info. So the left-hand picture here is the empirical distribution of this statistic across the whole UK at any given moment in time. And you can see that Medway is right up here on the very end. And then you can see how it sort of relates to, in the upper right here are sort of now drilling into all the individual postcode districts. So it's concentrated certainly over in these particular regions in this week. And you can also see adjacent postcode areas, and you can see that Canterbury and other areas of Kent also have elevated homozygosity. Now, this is lineage. The lineage which drives this, we can extract what is basically the thing we're seeing over and over again, is this B.1.1.7. We've been looking at this particular example in our sort of internal group meetings for a few weeks because it keeps coming up amongst the most homozygous. The hypothesis was there was some kind of a rapid transmission that generated this homozygosity, but we didn't really dive into it any more than that. And this lineage has actually become quite famous in recent days. And one of the things that we didn't appreciate, but which other members of COG had already been looking at, was that not only is this lineage spread quickly in this part of the country, but it has a lot of mutations. So it has 17 or something coding mutations. I think it's 14 amino acid substitutions and 3 coding in-frame deletions. And that's very unusual. I won't talk much about it because I suspect Eric might get into this. But it is an unusual lineage because it picked up so many mutations so quickly. And the final part of the story that I'll share is that we were then contacted by the Lighthouse labs that we're working with. Several of them use a 3-channel Thermo Fisher TaqPath assay. And this basically does 3 PCR reactions to test for presence of the virus. And the 3 reactions are in the ORF1ab gene, the S or spike gene, and the N gene. And usually all 3 are highly correlated. If you see a signal in one, you see it in all 3. But occasionally this you see a different pattern. And there was a query raised with us about an increase in the negative results in the spike PCR, so-called S dropouts. And one of the benefits of having done so much sequencing from this random flow of samples is that there was a set which I immediately had access to, which happened to have both the sequence and the status of this S dropout. It's a small sliver of all the sequences, but it had the key data. And we could confirm very quickly that over 99% of these S dropouts had a deletion of amino acids 69 and 70 in the spike gene. So it's quite clear that what was happening was this mutation, del 6970, causes the S readout of this assay to show negative, to show false negative. Just to state, this isn't really a concern in terms of the test validity, because there are the two other channels and all of the labs using it will call a positive test result based on 2 of 3. So it doesn't cause a false negative of the overall assay, but it clearly shows this sort of signal. So the question is, where is this del 6970? Well, if you look at sort of all the pillar 2 sequences over the last several months and ask what fraction have del 6970, you can see it was running at 1 or 2% for quite a long time, but it has risen very steeply in recent weeks. For those of you who are actually looking at the sort of live COG-UK data, I'll just note that the most recent PhyloPipe run goes up here until basically the middle of November. And these 2 data points are the 3rd and 4th weeks of November, where the sequences will sort of, they've been uploaded to Climb and they'll appear in PhyloPipe as soon as it runs. But they're all at Sanger, so I've confirmed 6970 is increasing in frequency. And the observation is that 6970 deletion is one of the mutations that is on this this particular B.1.1.7 lineage. This 6970 has arisen multiple times independently. So before this lineage, the sort of 1 or 2% back in the recent months was due to other lineages that carry this mutation independently. And in fact, if you now ask, of all the 6970 sequences from COG-UK over time, what fraction are this lineage? Well, you can see it's 0 back in September. So basically the difference between the dashed and the solid line is whatever other 6970 lineage is circulating. And it was pretty low, the new one, until essentially late October, early November. And then it has increased at a quite steady rate over the last few weeks. That's obviously going to be covered in more detail by Eric's talk and is the subject of ongoing investigation. To summarize the Sentinel surveillance piece, we think we should continue doing this, if at all possible, through 2021, because we think it still will have value. As I mentioned, we hope the new facility will increase throughput and decrease turnaround time. One thing that means is that if case numbers decrease to the levels last seen in the summer, obviously that's not happening right now, but as vaccinations begin to be deployed, it will be the case that we can sequence nearly all the positives that flow through these lighthouse labs. That obviously gives you very good surveillance, but it also means that for all kinds of other use cases, you get them essentially for free, because basically any sequence you go back and look for because of an outbreak, because of an importation investigation or whatever, it will be there. And we do think it wasn't the one we had in mind, to be honest, about biologically interesting mutations. We were thinking about vaccine escape really, but the contribution of these quasi-random sequences, because certainly the substantial majority of the sequences that went into the detection work that others have done to identify this spreading lineage came from this random sampling approach. So we think it provides proof of concept for the strategy. Thanks very much. And again, thanks to all of the hundreds of colleagues, both at Sanger and in COG-UK more generally, have contributed to the work.
Jeff, thanks very much. We must move on rapidly, but that's a Herculean effort, and showing us the scale of it was amazing. Now, the next talk is Judy Breuer. We'll be trying to finish about 4:20. We've got 2 more to squeeze in. I don't know whether this is a hypochlorous acid study as written, but anyway, over to you, Judy. Judy, are you there? If you're talking, you're on mute.
Can you hear me now?
Yes, perfectly.
Can you see, and can you see my screen?
And we can see the slides. That's great.
Excellent.
Can't
okay, well,
Yeah.
Thank you very much for inviting me to speak about the HOCI trial, which is a trial that's nested within COG-UK. So the basis of this you've heard about already. Hospital-acquired infection was observed to contribute at least 11% and probably as much as 20% of all hospitalized infection cases in the first wave. And it was also noted in the first wave that hospitals were a major source of infection spreading into the community, not only into care homes, but also from healthcare workers through their families into the community. So we've known for a long time, and Estee has actually touched on this, that sequencing pathogens and sequencing viruses is better at identifying the sources of infection and transmission routes than the infection prevention and control epidemiology alone. But it has always really been retrospective. And it's really with the advent of SARS that suddenly the ability and the will to actually do rapid sequencing has happened. And as you've seen from Estee's talk, it was reported from the first wave to improve the management of hospital outbreaks. So on the basis of that, we petitioned COG-UK to set up the COG-HOCI hospital onset COVID infection trial. And this is a phase 3 prospective interventional cohort superiority study. It's urgent public health supported, and its aim is really to gain evidence that sequencing really does make a difference in the context of hospital outbreaks. It's about, as somebody mentioned at the very beginning, future-proofing the enormous and excellent sequencing that has been going on through Pillar 1, sorry. So what we're asking is, does the availability of SARS-CoV-2 sequence data to the IPC teams reduce the incidence of hospital-acquired SARS-CoV-2 infection and reduce the number and size of hospital outbreak? And is the impact of sequencing on IPC practice dependent on time to result? And you've heard about the rapid turnaround that Estee has described. We were aware that not everybody might be able to achieve that, but we just don't know how important that is. So as I mentioned, this is nested within COG-UK. You can see we have sites all over England. We're very pleased to have Glasgow in Scotland as part of this, 14 hospitals, 11 COG-UK sequencing hubs. And just to sort of draw your attention to the fact that we have a large number in London, and those sites are now actually collaborating on an independent sub-study to look at the emergence of this variant and how it's affecting what's happening in our own hospitals. So the design of this was that we set up for 6 months. We aimed to have a baseline collection period where we would actually monitor what was being done for infection prevention control using standard methods, and that would last 4 to 8 weeks. And those patients would be entered into a database. And then we would introduce rapid turnaround sequencing, aiming for 48-hour turnaround, and we measure whether that changed infection prevention control practice and whether it achieved the aims of the study. We also included a slower turnaround time, perhaps 5 to 10 days, and that was to mimic the possibility that people would send sequencing away to a central facility, as might be indicated. And finally, there's a 3 to 5 month analysis period. So data collection is aiming to end at the end of April, and results will be reported from July to September of 2021. So there are 2 main tools that we have developed in order to run this study. One is a very, very comprehensive set of case report forms that capture really detailed information. And this is a huge ask for the sites. It's fortunate that we're UPH badged because it's quite a difficult study to run, but it is capturing really for the first time, I think, the sorts of things that are done when you have a putative cluster or hospital onset infection. And you can see there are several case report forms for each of the phases. And you can see that I've shown you the detail that's required on one of them here. And the second thing that we were aware of was that reporting the sequencing back to the IPC teams was going to be a challenge. SARS-CoV-2 is less mutable than many other RNA viruses. It's estimated that it's about 1 mutation every 10 transmissions. And so you may well get identical sequences that are not directly transmitted. And this is a slide borrowed from Estee's very, very nice paper which shows that you do require then to do an awful lot of collecting of metadata in order to make sense of whether samples are, whether there isn't a cluster outbreak and who is infected by whom and who's in clustering with whom. So we decided to take a slightly different approach, and we decided that we would develop a reporting tool that could be delivered back to infection prevention control teams as far as possible without the need to collect a lot of metadata after the sequencing had come through. And what this does is slightly different. It doesn't look at cluster analysis, which is what many people have been doing. It actually does a similar thing to what's going on, what Jeff Barrett was describing. It compares the sequence from a HOCI case, and we defined a HOCI case as being positive or symptomatic 48 hours or more after admission for the first time. And it compares the sequence from that case with sequences from all the other positive cases in the hospital and with community sequences. So we're saturation sequencing the hospital. And what we do is we integrate the data from the community. This is the pillar 2 sequencing largely carried out by Sanger. And that's we have the postcode and sequence from the community. We have the postcode and sequence data from the inpatients and also the local pillar 2 data. And we also have metadata from the hospital, and we're able to look at when people were admitted and which wards they're on, and we're able to match closely related sequences to others in the hospital. And all this is synthesized using a model, a probability model, to issue a report that gives you the probability that a new hospital onset COVID infection has been acquired in the hospital. Because of course we don't always know whether people have acquired it in the hospital, depending on how soon they acquire it after admission. And it also identifies a list of putatively linked cases and where in the hospital they are located. So this is the report. It's a one-page report that goes back to the IPC teams. It gives you the information about your focus case, your HOCI case. It gives you the sequences that are likely to be linked. And it also gives you a statement in red. It's the same form for everything, whether it's linked or not, or whether it's hospitally linked within the ward or linked within the hospital or linked within the community. One form serves for everything, and it identifies those patients that it thinks are linked. And we also sometimes issue a phylogenetic tree to just provide a visual reference for that data. And then we get a little diagram here. And in fact, this patient had been on that same ward a week earlier. So it provides a simple report that goes back to the IPC teams. So we were very conscious that this is a break from tradition. So we validated this using data from Glasgow and Sheffield, kindly provided by Emma Thomson and Tushin De Silva and their teams. And what we did was show that the tool is able to identify all definite and probable HOCIs. And it also identified an additional 15 definite and probable HOCIs that had not been reported by the teams originally because of pressure of work. It identified linked cases either on the ward or in the hospital for 92% of those definite or probable HOCIs. And it identified a linked healthcare worker in over 80% of transmission clusters. And that sort of accords with the some of the estimates that came through about the importance of healthcare workers in transmission during the first wave. Importantly, it was able to identify that 30 to 40% of indeterminate HOCIs, these are the ones we don't really know what's going on. They become positive very shortly after admission, and we just don't know whether they're community acquired or hospital acquired. It gives you a probability of hospital acquisition in 30 to 40% with a linked case. And it identified, in addition, mixed outbreaks on 4 wards and refuted the inclusion of 35 cases as part of ward outbreaks. And just to say that this is now, we're now entering the rapid feedback time part of the trial for some of the sites. And this is now feeding back full reports identifying linked infections within 48 hours, and very nice data coming out of Sheffield, who've managed to get that working. So just to give you a little brief overview of where we are so far in the study, it's been going for 2 to 3 months. Not all the sites were enrolled from the beginning, so we've enrolled the last one last week. We already have 358 HOCI cases in, all baseline. You can see that about 35% of them are indeterminate, so those will be quite interesting to look at. We're not reporting back at this time. This is just data collection to provide the baseline. And this is just the to show you that we've started to analyze data, and this is the age distribution of our HOCIs, which is probably in line with what is being seen nationally. So our deliverables will be whether or not SARS-CoV-2 sequencing results in quantifiable improvements in the control of hospital infection, or is it just nice to have the data, for example, for root cause analyses? The possibility of extending pathogen sequencing to for IPC practice to NHS hospitals beyond centres of excellence. There are many centres of excellence that are doing this really well, but there are very many hospitals that do not have pathogen sequencing, do not have the expertise to do this. We're evaluating how well this could be used in those. We have at least one DGH and several hospitals have never used sequencing before. And this is a nested qualitative study that's going on. We're asking what is required to achieve NHS-wide pathogen sequencing for infection prevention control. And we're doing a cost and cost-benefit analysis with a nested health economic study. So I'd just like to finish by acknowledging that I couldn't fit everybody who's contributed to this study on the slide. Just like to highlight the COG-UK team, Sharon, who sponsored this study, the UCL team who've done a lot of the development of the clinical trial unit, Emma and Tushan and their teams, James Price and his team in Imperial, who contributed to the development of the study tools and study material. Thanks very much, and happy to take any questions.
Thank you very, very much, Judy. So Gordon Dougan is going to summarize now what were, to my estimation, an outstanding series of talks. So over to you, Gordon.
Yes, it's possible to give me power to share the screen. I've got 2 slides. I don't know who's got that.
I've stopped sharing mine, I think.
Okay, I think I should be there. Okay, I think, can you see that?
Yes, very clear.
Okay, so we've only got a few minutes, so very quickly, I think one of the big challenges with COG-UK was when we set up, we had no plan really of action of how to do it, and how do you drive policy from that point of view, and how do you drive innovation? So basically we started off by just adding groups as they came along, but by around May time, it became very clear that we had to have some kind of a simple organization, not only for in terms of governance, but also for data management, because this data was very sensitive, and also for generating sequencing and providing access to that sequencing. And so all of the different delivery groups sit over here, whether that's Sanger or whether it's the different dispersed groups in the different countries of the UK or sequencing centres. We then built up a management team, which also was involved in developing the SAGE reporting systems. But the crux of COG-UK is the steering group, which is a joint steering governance structure where we have all the representations from the different key technologies in the organisation, but also from the devolved governments. And that really is a focus through which all of the information goes through to the chair. And we get then a single voice that then determines policies and communicates policy through to the government. So this structure seems to be working pretty well. And we've got a sense check group over here, the advisory group, who are not directly connected with COG-UK, but are SAGE individuals who are very experienced. And Sharon and Ewan, who runs the management team, can go and talk to that group, and they will also come in and check if any issues arise around governance. So that's basically how we manage the consortium, and that's how policy is determined and then communicated through as a single voice. It avoids lots of duplication of messages across from the different components, and it really is a way of channeling information. So moving on from that slide, if I can do that, you then have what challenges we've got now, and some of these challenges have been summarized really well in this last session, really. And you've got to sort of, if you like, to balance the policy as a service side and delivery side of this data, which has to have impact and has to be used to intervene in the disease, with the ability to do research and innovation. And sometimes these two sides don't mix so well together. In industry, you tend to have a research separated out from the delivery of projects in terms of development and project development teams that then deliver. So what are the challenges that have emerged really from this session? So we've heard about sequencing analysis in real time. One of the issues that we have is that, for example, much of the Sanger sequence that's been done has been done on older samples. Can we get more in information in real time? What are the challenges really around getting sequencing of high quality? Andrew mentioned the issues around quality of sequence, and we, at least in the UK, we're well versed in the need to have very high quality sequence analysis, and with MRC Climb and all of the health checks we have there, where we can manage that to some extent. Then we've often discussed the need for local impact. Judy mentioned they're looking in within hospitals, but often some of the most impoverished communities are the people that get less access to this kind of information in terms of having impact on the management of disease spreading in those areas. Ben mentioned, for example, some of the behavior of students who are high risk, whether they've got cycling helmets on or not, but they are high risk, and you can track the spread, the spread of infection, which is driving infection within that community. I think another big challenge, which also aligns with the two boxes, is the culture of a delivery service, which is very much bureaucratic and is meant to, and drives out innovation, compared to a more innovative sequence, research-driven ethos. And I think what we're going to have to learn in the future within COG-UK is how to balance those tensions to keep as productive as possible. And the last and final point, really, I think Patrick mentioned this right at the very beginning, and I was very pleased he did, is that COG-UK is really a model, not, I don't think just for this virus, it's a model for many other diseases. And how do we translate, or should we translate COG-UK into these other diseases, and how might we do that? And it's something I've been very interested in. I gave a lecture at PhD, the Terberg lecture 10 years ago, where we were advocating this, and I'm very encouraged that we're emerging from COG-UK as a system whereby not only can we get better by applying ourselves to these challenges on this box, which have been highlighted by the different presenters, but also as we go into the future, can we address other diseases? And that's really my summary. Thank you very much. Maybe caught up a bit of time.
Gordon, thank you very much indeed. Now, I think the chat has been very rich, as people say, but there's a lot of information there. I think that we don't really have time for questions, which is a shame, but the talks ran on and the chat is there. My instructions are you can have a break until 16:25 at the very latest. And then Ian Young, my equivalent in Northern Ireland, will do a better job of chairing and timekeeping. But it's been a real privilege to be involved with this session, and go and stretch your legs, and back 16:25. Be there or be square. Thanks very much.
Thank you very much.
Hi everyone, I think it's time that we should get on with the next session. So you'll probably, the keen-eyed among you will probably notice that I am indeed not Ian Young. So Ian was called away at the last minute for an urgent meeting. So I'm afraid you're going to have to put up with me chairing this session. We've got some great talks though. The sessions on mutations, implications for transmission, disease severity, therapeutics and vaccines. And our first speaker is Professor David Robertson from the University of Glasgow. David, can you take the helm?
Yeah, let's see if I can get the share working. Is that working?
Perfect, thank you.
So I was going to talk a little bit about mutations and I guess in some ways give an introduction to this session and I guess try and convince you, I guess, that although this virus is evolving slowly, we do really have to take the surveillance very, very seriously. I think, the number that we're shared all the time is the 2 mutations per month in terms of average rate of evolution. And it can be kind of misleading because when you look at next strain and it's all temporally represented, everything spreads out quite nicely. But if you take away that temporal signal, there isn't, I think that shows there isn't masses of evolution occurring. This is a very slow evolving virus. And that seems to be very much its mode of operation. It's specialized to be very successful in bats by retaining this property of being not species specialist, but really looking at specializing on horseshoe bats, and all that allows it to jump between these species very readily. And it's really important to appreciate that most of the mutations we observe aren't actually replication errors. We think these are probably antiviral host molecules that are introducing errors into the virus RNA. And so this is really a very slow evolving virus. And when we looked at the first phase of the pandemic, it was really very clear that there wasn't an awful lot happening. There's not an awful lot of positive selection, and there's a few sites that show some evidence of change. And that, I guess, really that represents the fact that what's emerged out of the bats, really with probably very little change, is a virus that's been very successful in the human population, highly transmissible, and so really just at the get-go able to spread very rapidly, and of course cause the pandemic. And then as things have developed, it has become clear that although on average there's not that many mutations occurring, so we've observed about 4,000 different independent events in spike, out of whatever 250,000 genomes. There are a few now that are kind of accruing some significance, and so D614G is the one that might let us actually call this a different strain. And I think Eric might talk about this a bit, but it's clearly gone to fixation, and that's helped by it emerging quite early in the pandemic. But also, it's really it is slightly more transmissible, and all UK sequences now contain that mutation. And then there's a few others like the A222V that's emerged in Europe. Again, that seems to be sweeping into the UK at high numbers. And then I'm going to talk a bit about N439K. That's one we've spent quite a lot of time studying. And it and a few others now are starting to occur in the receptor binding motif. And of course, this new one, N501, that's part of this B117 lineage that's causing a bit of a fuss. And so N439K was very interesting because it emerged in Scotland in March, and it went to one person in England. And so that's this lineage 1. And what that gave us, I guess, an opportunity to do was characterize it in great detail, because it really arrived on our doorstep in Glasgow. And then as we tracked it, really it was quite fascinating because with the lockdown, essentially that lineage went extinct. And as we were working on this paper, we had noticed that this other lineage started to emerge with this N439K replacement here. And now this is actually now kind of quite a relatively global lineage. The last count is about 26 countries. And so it's very much sort of on the march. And what's happening really is, and I guess this helps to sort of, this mutation isn't in the similar way to D614G making the virus more successful, but at the same time, it's not making it any less successful. And so when we look at the phylogenetics and growth of this virus lineage, and so we do this in Scotland because we have so many sequences, it's not atypical. It's not increasing in numbers any more quickly, but at the same time, it seems to be relatively successful. And it's just a little bit worrying. The fact that the virus can accommodate what is a relatively interesting change is something we have to be concerned about. And when I say interesting, if you look into the receptor binding motif, so the part of spike that binds to ACE2, K439 is actually making a new salt bridge, and so that's replacing the need for K417 and moderately increasing the binding then to the receptor. And so really what this is showing us is just how adapted this virus is to holding on to ACE2. It can accommodate changes in this kind of phenotypic plasticity. The structure is accommodating changes that allow it to hold on to the ACE2 receptor more strongly. And in itself, that's maybe not something to be concerned about. But what that does then permit is other mutations that may then have otherwise been a little bit deleterious, or might then have more significance in terms of antibody escape to accumulate. And really, I think this is the bit we're starting to see occurring now. We're starting to see mutations. So N439K accumulate is on the background of D614G. And now, interestingly, there's a deletion that seems to be co-occurring with this one. And so you have this really highly malleable or plastic region of the virus that allows it really to accommodate change. And so far, we've had no vaccine pressure. So, we're not seeing anything, but I think we have to worry about the potential for change. And this is just some images from Andrew Rambau about showing that deletion. And so in that second European lineage, the second European lineage 2 essentially had a fixation event. And so that deletion now, so 439K emerged here, and then the deletion here. And so many of the circulating viruses now have that deletion co-occurring with that receptor binding motif change. And then, of course, we had the mink outbreak. Again, we have an RBM change, increasing affinity, and then a deletion. And so there's no particular evidence that these are direct interactions. These are quite distillant in the structure, but it does show that the virus is starting to accumulate changes that are independently potentially important. And then Ravi Gupta has been looking at this too, looking at 501 here. And so you've got actually the deletion being the one that's occurring most with these different receptor binding motifs. And at the moment, it's really, I think, not that clear why that is, but there's clearly some functional or potential functional difference here that's going on. And so with Will Harvey, we've been looking at these mutations in the context of the protein structure. And so when you look at spike and you look at accessibility, so how easily an antibody can get to a residue, you can see that the ones that are seem to be escaping antibody in some way are here more accessible. Then you have this other region here of the spike molecule, and then 614 over here, so not really contributing. There's no evidence it contributes to escape as you'd expect. And importantly, when you look at mutation frequency and you highlight on the spike molecule, we're not seeing that many changes in those accessible regions. But that's because there's not really been any population level immunity selection pressure put on the virus. Most of us are susceptible. And so it will only be really when viruses are used that this will start to change. What's the next? And so this, I guess, leads us to the sort of the importance of this, because now with this new B117 lineage, not only have we had observed receptor binding motif changes, we had observed the deletion, you now have a set of other mutations all co-occurring in the spike molecule together. And so although there's no reason to believe this will generate a vaccine, these will escape vaccines, what it's demonstrating is the virus's ability to evolve relatively quickly. So given that background of slow evolutionary rate, in the important region of the molecule where most of the vaccines are going to target, most of the antibodies, are going to target that are elicited by the vaccine, we're going to observe change. And I think it's quite Sebastian's visualization of those 17 changes on the spike is a little bit terrifying to see all that change relative to the Wuhan strain, which the vaccines have been designed against. So really, I guess it's important to remember that we're really looking at virus that's emerged ready to go. It's very unusual. Viruses usually, because of the intrinsic dependency on the host, are fairly wired into the species that they've kind of co-evolved with. And so to have a virus that's able to jump to one species, to humans, to mink, and cats, to ferrets, to different animals, and pangolins, of course, and be successful in the different species is really something concerning. And so that generalist nature is really the consequence of that adaptation to broadly to ACE2. And so when we see increased binding, or that's phenotypic plasticity. That's just some wobble in the ability to use that, and that would be important because it could permit other mutations. And then that's what we're starting to see. We're starting to see D614G, then receptor binding mutations, the deletion, and then multiple other changes. And really, I guess what this outbreak now in England, this new fast-growing lineage is showing us is the importance of surveillance. When we have everybody vaccinated, if a vaccine escape mutant emerges, we're going to have to be able to turn that surveillance around much more quickly than we're doing at the moment. And I think these are all lessons we're getting at the moment, ways to ways to sort of maybe to change how we operate, to have procedures in place that will allow those kinds of responses. Oh, and so many people involved, and I think this is, as I think many people have echoed already, this has been an incredible consortium, and also working just broadly and internationally with different people. I've never sort of experienced so many people pulling together to try and make a difference. Thank you.
Thank you, David. That was really excellent and a really good summary. Set the session up very well from the beginning. So our next speaker, we're going to move straight on. Hopefully we'll get a few minutes at the end for some questions, but if anyone has any questions for David, please post them to the chat and hopefully David will respond. So our next speaker is Dr. Eric Volz from the Imperial College London, and he's going to give us a talk about the epidemiology of SARS-CoV-2 genetic variants.
Thank you, Andy. Let me know if you can't hear me okay.
We can hear you fine. Maybe if you put your presentation on full screen so that we can see the full image.
I think this is actually the best I can do at the moment.
We'll go with it like that.
I'm sorry about that, but so thank you. This talk will be a bit more on the quantitative modeling side and what we can learn in the early stages when a variant is starting to emerge. And it's also a bit of a cautionary tale because it's easy in the early stages to see misleading signals. When a variant is emerging, epidemiology tends to lead a little bit our virological knowledge. So we might see growth of a variant where there wasn't much preceding evidence that it would have much of an effect. And in that situation, we have to be very careful. In particular, I'll show what we can learn about the growth rate of a variant and what it implies about a fitness advantage. So we immediately want to know if a variant might be more transmissible, also how it's spreading geographically, and then finally if it might lead to different patient outcomes. So is it associated with greater rates of hospitalization or death? And to illustrate this, I'll show two things. Most of the presentation is about the variant A222V, which came out on the background of global lineage B1.177, originating late summer this past year, but spreading fairly quickly in the UK in August and September. And then I think many people are interested in this new variant N501Y, and especially in the way it occurs on the background of lineage B117. So I'll make some comparisons and contrast that as we go. So regarding this A222V variant, this first got a lot of attention when Emma Thomson and co-authors put up a preprint showing that it had likely originated in Spain and spread very quickly, in some cases reaching a majority of all sequences in a variety of European countries. And by this point, it was also increasing very rapidly in the UK. So the obvious question in this case is the change in frequency reflecting a fitness advantage, or is it just due to chance? So, and by chance, we basically mean genetic drift and founder effects. And we can certainly expect founder effects to be playing a large role in this case because of the large amounts of holiday-related travel from the UK to Spain during the same period that it was increasing in frequency. So we do know that founder effects is going to be a major confounder in this case, and we need some way to adjust for that. So fortunately, due to our recent experience studying the D614G variant, we had some methods in place to study exactly that problem. Founder effects was also a major confounder with that variant. And the basic idea is to do some phylogenetic corrections. We can identify parts of the global phylogeny which likely represent distinct introductions into the UK, and then we can study those distinct introductions side by side as a comparable set. And if done correctly, and if we have all the data we need, then this should adjust for the fact that if, for example, this green variant, these green branches represent A222V, that we'll have more of these introductions at certain points in time. So here's what we get when we look at the change in frequency of A222V over time on the left-hand side. And this was the state of our knowledge as of early November. And what's shown here is a plot of the log odds ratio of sampling this variant among all COG-UK sequences over time. And there's a theoretical reason for wanting to plot it out this way. If the variant did have a constant selective advantage, we would expect its frequency to change like a logistic function, and all these points would fall on a straight line. And you can also very easily read off what the selection coefficient would be as the slope of that line, corrected for the generation time. So when we look at plots like this, we see basically two phases of growth, a very rapid rise in late summer. We think that that's corresponding to multiple imports from mostly holiday travelers, followed by a second phase that might putatively have represented some selective advantage. So before moving on to what happened next with A222V, we can contrast this on the right with what we're seeing currently with N501Y variant. So we're still basically in the very early stages. We basically have 1 month of growth for comparison, whereas we've now observed A222V for more than 3 months. And the growth rate does appear to be quite a bit larger with N501Y currently. I'll come back to that in a moment. So back to A222V, back in early November, I was actually willing to bet that there was some small selective advantage and that this would eventually complete a sweep in the UK within a few months. And I was wrong, because over November, and this is coinciding with lockdown, the frequency leveled off. And in fact, the selection coefficient currently is pretty close to 0. So this is part of the cautionary tale that trends you see early on don't always pan out. It is important to let more data accumulate. Also, that the reasons for this leveling off are not exactly clear, but we do now know that there was quite a lot more travelers in October than I had previously known about. And so travel didn't fall off as much in October as I originally expected, and that's probably a very large part of the explanation. We can model this a bit more quantitatively by fitting a logistic growth model. And the results of that are shown on the left for A222V and on the right for N501Y. The main point of A222V is that we get a selection coefficient of around 15% using this method, but the model fit is not particularly good. There are lots of outliers early on, and there are lots of outliers quite late. So we wouldn't expect that a logistic growth model is necessarily appropriate in this case. With N501Y, it's really too early to tell, but this is the current state of our knowledge. We know that it's growing very quickly, we know that the sampling frequency is very noisy and over-dispersed, and this model is not currently accounting for that. But from what we see so far, it is growing very quickly. It's growing faster than A222V ever grew. So it is important to keep an eye on this. Finally, I want to make some observations about finding associations between genetic variants and severity. Since there are a lot of papers popping up currently, especially using GISAID data, showing associations between different variants and the probability of a fatal outcome. And there are lots of pitfalls you can fall into when you carry out such an analysis. I think we have a pretty good example, having done a very detailed analysis of A222V. So very early on, October, November, we actually saw a significant increase in the probability of a fatal outcome for patients with the A222V variant. And that was spurious. It was very likely due to correlated and biased sampling at the time. And we very wisely decided at that point to wait a few weeks before escalating it. And indeed, when we waited a bit longer, most of the signal washed out. The figure on the left shows a survival curve comparing the mutant, in this case A222V, and the wild type. And there does appear to be a little bit of an effect, but this falls short of statistical significance. Another slightly odd feature of this variant is that we seem to see different rates of hospitalization. So if we compared over time the probability that someone would end up in Pillar 1 or Pillar 2, Pillar 2 being community sampling, Pillar 1 being mostly hospital sampling, we saw that there was much higher odds that someone would be sampled in community testing. And these results were actually contradictory. So it seemed like A222V carriers were less likely to end up in hospital, but more likely to have fatal outcome. And it's only recently, as data has accumulated, that these contradictions have been resolved. And again, it is mostly due to non-random and very noisy sampling. Another clue was when we broke down the analysis by region and showing quite large differences between regions, which does not make any sense if there was a biological explanation for these differences. But then only once after correcting for different rates of sampling in different places and also using the phylogeny itself to adjust for correlation between outcomes that we found a more consistent outcome between regions. So at this point, we really don't think that this variant is causing much of an effect on hospitalization or on mortality. So that's it. Thank you. There are too many people to acknowledge, so I haven't attempted, but thank you.
Eric, thank you very much. Really interesting stuff. Again, if anyone has any questions for Eric, can I direct you towards the chat? We are running a few minutes behind, so we may not get a chance for questions at the end of this session. So our next talk is from Professor Emma Thomson at the University of Glasgow. And Emma's going to be talking about mutations and clinical consequences, which follows on nicely from the end of Eric's talk. Emma, please take it away.
Hi, thanks very much. Can you see all right? I'm just going to stick it on slideshow.
Yes, we can.
Great. Thanks very much. So the title of my talk is on mutations and clinical consequences. And so I'm going to touch on what all this might mean for us in terms of outcomes and the importance and clinical relevance of these mutations that we're seeing. If I've got time, I might just also highlight some of the pitfalls that Eric was mentioning, and I think are really important that we address so that we interpret clinical data correctly. I'm going to talk mostly about the N439K that David mentioned, but concentrating on the clinical sort of aspects of that. But this is obviously applicable to other mutations that are rapidly evolving, of which there are many. Just to start by saying that many thanks to all the collaborators who've been involved with generating the data on this. It's been really, I just like to reflect the everything that's been said about the kind of enabling of science, and particularly would like to thank Sharon Peacock for enabling us to work together with many people as part of the COG-UK consortium, but also the links that we've established now with people internationally as well as across the UK has been really extraordinary, I think. So I'd just like to highlight some of the sort of critical clinical endpoints as I see them, and other people might have other views, and I think that this is something that should be discussed at length for monitoring mutations in the SARS-CoV genome. I think perhaps one of the most topical areas that we really need to be right on top of at the moment is escape from vaccine-induced immunity. Of course, we are now rolling out vaccination to high-risk groups, and so this is going to provide a very strong selection pressure in the population, and we may well see a rapid rise in mutations as a result of this. And of course, we need to look for any evidence at all of escape from vaccine-induced immunity. Also, we need to look out for reinfections or escape from natural immunity that again might arise as a result of changes in the genome. We've touched a little bit, and Eric touched on the possibility of mutations affecting the clinical severity of disease. I don't think we've seen any evidence of that happening yet, although certainly there are changes which are associated with higher viral load. So we may expect to see changes in clinical severity, but perhaps less likely than escape from vaccines, for example. I think we also need to look out for mutations that affect treatment efficacy, and particularly treatments such as monoclonal antibody treatments or other, for example, like polymerase inhibitors like remdesivir. And then mutations that alter transmission dynamics has been covered really in a lot of detail. I'm going to highlight just again the plasticity of the receptor binding motif. It really is a sort of a key to understanding the kind of problems that we might expect to arise clinically. And the receptor binding motif, which is the area that binds to the ACE2 receptor and is likely to be the target for neutralizing antibodies, is quite plastic, and that's really important. And just look down here at the N439, which we've heard a little bit about, and I'm going to go into a little bit more depth on that. And then also, for example, the N501 sits just above, and there are other sites as well which are plastic. And so we can expect to see changes in these areas, and we certainly need to keep our surveillance up for changes here. David mentioned, I won't go into too much detail on these lineages, but just to highlight again that there have been two main lineages associated with the N439K mutation, and this is how it breaks down in the UK, in Scotland and England, and then in the Republic of Ireland. In fact, there's been a lot of this particular change forming the majority of sequences. It's important, as David said, because of the issue around the formation of a salt bridge that strengthens the ACE2 receptor binding motif interaction. And this actually could have been predicted by earlier work on SARS. SARS-1, this is where the equivalent site does form a salt bridge. And when the N439 changes to a K, it almost certainly results in a salt bridge, thereby stabilizing the interaction between the virus and its receptor. We know from binding assays that indeed this change increases binding. Now I'm going to talk a bit about whether or not this has clinical significance in terms of severity of disease, which is something that we should be following. It's quite tricky. So we have this information from the Scottish data, and just because the N439K actually arose within NHS Greater Glasgow and Clyde, where we happen to be doing pretty detailed surveillance on clinical outcomes of our participants in a prospective manner. And I think I would advocate for Sentinel surveillance sites to be able to look for clinical outcomes in people who have these mutations. However, what I'm presenting now is a result of an effort of maybe around 40 people gathering data on around 2,000, almost 2,000 patients. So we, as has been noted, we saw this sort of rise in cases of N439K before the first lockdown in Scotland. And we looked at clinical severity and created our own clinical severity scale, which reflected hospitalization. And we've been talking a little bit about limitations of understanding clinical data. One of the major sort of confounders with the first peak of infection around the UK was that we probably all initially, as infectious diseases physicians, hospitalised people who were found to have COVID infection, whether or not they had a severe infection. And that seriously biases the WHO ordinal scale, for example, which includes hospitalisation as a measure of severity. So we modified that and used oxygenation status to look at whether or not people had more severe illness. And what we found really was that across all the age groups, as we've seen multiple times in other studies, there's a huge age differential in terms of risk of mortality. But we didn't see a significant difference between those people who had an N439 or a K439. However, that virus is certainly just as virulent. We then looked at viral loads. So we found that viral load was in fact higher in people. So this is CT value, so lower CT meaning higher viral load in people who had this mutation, the N439K. And this was significant, and it was significant beyond the effect seen by the D614G in isolation. This is just to show that CT correlates very strongly with viral load when measured with RNA standards. And then we grew these viruses in culture. And again, this is one of the sort of proxies that I guess we can do to try to find out whether or not the virus is more capable of replicating rapidly. We did find that there's a slightly increased takeoff of the 439K in culture. And we find the same thing using in a cross-competition assay using next generation sequencing as an output to quantify the number of Ks versus Ns in cross-competition and culture. When TMPRSS2 is overexpressed in cells, you do see quite a strong effect of 439K taking over. We then looked at serum samples from people who've been infected with SARS-CoV-2. And in blue here are people who've been infected with the wild type virus, and you can see that there's decreased binding to the 439K receptor binding domain in this slide here. When we looked though at people who'd been, a small number of people who'd been infected with the 439K virus rather than the 439N virus, we found that binding was not particularly reduced, and I guess that does indicate perhaps subtly that if we were to tweak vaccines, if we do find there's a problem with vaccines in the end, tweaking them may well be possible. This is just the full change. And then we also looked at binding to monoclonal antibodies, and we looked at over 100 monoclonal antibodies. This was done by our colleagues in the US. And they found also decreased binding to the receptor binding domain 439K with monoclonals. And these are some of these monoclonals. And I'm just going to finish now with highlighting these different monoclonal antibodies that are in current trials. And so this is quite important for patients who have severe infection. They may be offered, for example, the Regeneron combination antibodies. And what we have found in our study is that one of these antibodies, 10987, binds less well to the 439K, and in fact also it neutralizes less well as well with pseudovirus assay using a VSV pseudotype. However, other antibodies seem to work okay, and this is the Lilly product and the Vir product. So it may be that sequencing could become important in terms of deciding what kind of treatments may be best for our severely unwell patients. And I'll finish there. To summarize, it's going to be important to look at key outcomes, for example, infection in vaccinated individuals as the vaccination is rolled out. Any lack of response in treated individuals, for example, particularly with monoclonal antibody treatments, which are where it's happening now in terms of new treatments. It's going to be important to consider clinical severity of disease and, of course, transmission dynamics. This is a huge list of co-authors on this paper, which is currently under review in Cell. Thanks very much.
Emma, thank you. That was really excellent. Again, if anyone has any questions for Emma, I'm sure that she'll be hanging around for a little while and we'll be able to answer them in the chat. Our next speaker is Professor Ravi Gupta from the University of Cambridge, and he's going to be talking about some SARS-CoV-2 mutational escape from therapeutic convalescent plasma. Ravi, are you there?
Hello, yes, here. I'm just about to share screen. There we go. How's this? Can everyone see this?
Yeah, we can. Perfect. Take it away.
Perfect. Okay, thank you so much for the invitation to speak, and I'll be telling you about some work that has been recently pre-printed, where we have shown probably for the first time the effect of neutralizing antibodies on direct spike-mediated evasion. So the case rests on an individual who was diagnosed with COVID-19 in May of this year. He had the first positive PCR result in May and actually was discharged after his episode of neutropenic sepsis, then presented with a persistently positive SARS-CoV-2 PCR and deterioration with breathlessness and cough. Required a bit of oxygen in intensive care and then came out onto the ward. And really over the next 6 weeks was relatively stable, but persistently positive for SARS-CoV-2 in nose and throat swabs. And in the second week of August, unfortunately, deteriorations set in with multi-organ failure and rising inflammatory markers. And despite intubation and treatment with tocilizumab, he unfortunately died. Now, during the course of that admission, he received a total of 3 bags of convalescent plasma from 3 individual donors in red bars, you can see here. And in purple, these are 3 separate courses of remdesivir of 10 days each. He also received dexamethasone here in June, and as I said earlier, a course of tocilizumab and later on prednisolone. So down here you can see the CT values over time. You can see really there isn't a pattern. There doesn't seem to really be any significant sustained downward trend in the CT values. And we had a number of different samples tested, not only nose and throat, but we had a number of paired ETA samples later in disease when the individual was intubated, and we had a stool sample as well. So we really were interested in the fact that we had available a number of longitudinal samples. So the samples that were taken were in a pipeline and sequenced by Ian Goodfellow by nanopore technology. So these 23 samples spanned 100 days, which are represented in this tree in green. The individual here is green. What we've also done is placed 3 other control patients into this tree, which is a UK tree of full genome sequences from the GISAID database. And these other individuals that are serving as controls are people who shed virus for between 30 and 60 days. You see that there's significant living about here compared to the others. This tree also shows you that the samples are unlikely to have been due to contamination or superinfection events because they all cluster within the same patients. We did some Illumina sequencing as well of each of these samples because, of course, nanopore is excellent for generating consensus sequences, but less accurate at a lower variance. And this graph shows you how Illumina compared to nanopore in a subsample of 20 sequences taken from the individual. And you can see in general the correlation is quite good. So along the bottom, you have some key mutations and spike that were observed in the patient that we'll go on to later on. And you can see that what the variant frequencies are by nanopore versus Illumina. And they're either usually very low or very high. But there are instances where there is a significant difference between the Illumina variant calling and the nanopore calling, for example, here for this 6970 deletion. So we validated really the outputs of the the nanopore sequencing through Illumina, even though we use the same amplicons. But we wanted to do this because part of our interest was in low frequency variants. This is a highlighter plot that looks at the whole genome and changes in a sequential order over time. Here on you've got the day since the first positive RT-PCR going down to 101. And we've generated, these are all consensus sequences generated from the nanopore data. And you can see that there's very little or no variation in the first 60 odd days here at consensus level. And then we start seeing changes in the virus that accumulate and actually shift quite dramatically. Certainly at day 93, 95, you can see there's a very different looking population there. And then you go back to a population with the 6970 deletion, which is here in black. So we found a number of interesting mutations that came up in spike. And what was really interesting is that they seem to be either there or not there in most cases. So meaning that the green here, as you can see, this represents 6970 deletion that we've already just talked about, and a number of people have alluded to, including David. It came along with this D796H mutation in S2, and that's shown by the purple line here. So both of these variants reached very high frequency. We did not have sequences available between day 66 and day 82 when this double mutation was first detected, but it was temporally related to 2 units of convalescent plasma that were administered on day 65 and day 66. And the kinetics seem to fit because the convalescent plasma has a half-life of perhaps 2 weeks, and this time point is around the 2 week time, period. And of course, then we see a dramatic drop-off during sequential samples, and interestingly, a replacement of that double mutant with a group of viruses that have Y200H and T240I. These viruses are presumably replicating in a situation where there is very low level of convalescent plasma. And then remarkably, we see a completely different population here defined by P330S and W64G in the spike protein. And at that point, the third dose of the third course of remdesivir was administered, and 2 days later, a third a bag of convalescent plasma. And you can see here that coincident with the convalescent plasma administration, over the next few days, you see increasing proportion of the variants that we saw right at the beginning following the first 2 bags of convalescent plasma. And notably, between the 2 green peaks, you can see that the frequency does not go down to 0 for the entire period. So you're seeing that they're present at low levels between the 2 peaks. And that's important because, of course, before, in a number of time points that we sampled before the convalescent plasma, there was no detectable D796H or Delta 6970. Here's a structure of the spike protein. And you can see here, up at the top in S1 is the Delta 6970. It's out in an exposed loop. There are no structures of what the deletion does because it's in a disordered area that's poorly characterized. The other mutation, D796H, is down here in S2. Again, poorly characterized. It's in an exposed loop in itself as well. And the first sort of impressions we had of these mutations was that potentially they might alter antibody binding and potentially change susceptibility to antibodies neutralizing in the convalescent plasma. So we set out to phenotype these mutations or characterize them further. So we generated pseudoviruses, which were based on lentiviral vectors that are pseudotyped with spike-expressing plasmids, and we made mutants. We made in this presentation, I'm going to show you data for the wild type and the combined D796H and 6970 mutant, and you can see here that we've measured the IC50 dilution for neutralization of our pseudoviruses, and essentially you can the taller the bar here, the more potent the sera are against the virus, and you can see here that CP1 was actually quite potent, and there was a significant difference between the wild type and the double mutant here. You can see that there's a loss of neutralization, significant loss of neutralizing capability of that first bag of convalescent plasma. If you look at CP2 and CP3, these were much weaker neutralizers of virus, but nonetheless, we saw significant drops in neutralization sensitivity of the double mutant that was observed in the patient during those 2 peaks that I showed you earlier. We also had serum from the individual, so we were able to obtain serum samples from in between the convalescent plasmas, and again, low levels of neutralization as you'd expect, and consistent with other studies, because of course the convalescent plasma has been diluted out, and we saw a significant drop, certainly for day 64 sera, when you compared the wild type to the virus. Down here in the table, you can see the fold changes, and they represent anywhere between 2 and 4-fold loss of susceptibility. If you look here on the right side, these are 5 unselected sera from convalescent patients from the bioresource, and we randomly selected them, not based on any particular criteria. And you can see for 4 out of 5 of these, there is a reduced susceptibility to those sera by the mutants as compared to wild type. The panel down here is demonstrating the fact that when we generate these pseudoviruses, the amount of RT activity in them, in other words, reverse transcriptase is one of the enzymes in the pseudovirions, and it's a way of normalizing your input virus. And you can see here that both viruses, the wild type and the double mutant, produce similar amounts of virus in the supernatant. And when we measure infectivity on target cells, we can see here that the 6970 double deletion is in fact a little bit more infectious than wild type, and it's around 50% more. So we've done a few more experiments in order to disentangle this a little bit further, and you can see here we've made single mutants, the D796 alone, and then the Delta 6970 alone, and then the combination that I showed you earlier. And as we suspected, the D796H confers a fitness deficit of around 50% of the virus, and the Delta 6970 is around 2-fold more infectious in a single round pseudotyping system than wild type. And when you bring the 2 viruses together, you get this sort of intermediate level of infectivity, consistent with the notion that the 6970 is there partly to restore infectivity to the virus, although it may have other functions too. Just on the right here is just infectivity data at different dilutions to show you that at every dilution, the 6970 is more infectious than wild type. So looking at the frequencies of the mutants that we observed in the GISAID database, you can see that all of them are very low levels except for the 6970, which has already been discussed and is the subject of ongoing work. On the right here is a radial tree of around 10,000 sequences. The red ones have the Delta 6970 double deletion, and you can see with 439K sequences, but you can see that 6970 does appear in different parts of the tree in clusters. So consistent with what's been observed in the UK, that 6970 can emerge on its own, or it can emerge on the back of other RBD mutants. So this is probably the first real-time documentation of what we think is virus escape from antibody. We provide genetic evidence, which is the fact that by deep sequencing, we're able to see an increase in a double mutant that is temporarily related to convalescent plasma. And not only does it arise during following convalescent plasma, but it then disappears during the washout period. And then when a third unit is administered, we see the return of that strain. We then show phenotypic evidence that this double mutant is less susceptible than wild type across multiple donors, and not only the different convalescent plasma donors, but also a panel of convalescent sera that we had. And finally, in terms of infectivity, we I think make this the important observation that 6970 does increase infectivity spike protein pseudoreceptor viruses. We've done this at least 3 independent times and made sure that this is real, but we haven't done this with full-length viruses in a replication competent system. So in our case, we think that the Delta 6970 compensated for loss of infectivity, although it may have had some role in antibody escape, and we're doing experiments to see whether the 6970 confers any difference in susceptibility to the sera that we have and the convalescent plasma in the donor. So in conclusion, we can say that the broad change of susceptibility suggests that individuals may have antibody responses that are outside the RBD and that may have resulted, for example, in the D796H mutation. I'd like to acknowledge a large group of people because although this was a single patient, it was certainly a large effort, certainly, and facilitated by the fact that COG-UK has done such an incredible job of sequencing. Ian Goodfellow, who provided not only expert advice, but also samples for resequencing. And of course, members of my lab and Cambridge University Hospital Trust, and my colleagues at UCL who are collaborators on this project. And finally, the Cambridge University Bioresource. Thank you very much.
Ravi, thank you. That was really, really great talk. Once again, we're running a little bit long, so there's already a question for Ravi in the chat. So if you could take a look at that, Ravi, and respond in there, that would be fantastic. So bringing up the anchor leg for this marathon showcase, the last talk before the closing remarks will be from Dr. Alessandro Carabelli of the University of Cambridge, and he's going to be talking to us about how we're coordinating mutation data for public health use. So Ravi, if you could stop sharing your screen, and then Ali, share yours.
Sure, thank you.
Thank you.
Can you confirm you can see it?
Yes, we can see it, thank you.
Okay, so thank you, Andy. So first of all, I'd like to thank the organizer and Sharon for putting together this incredible showcase. And hopefully I will hold your attention, even if I'll be mainly focusing on the coordination of the activities behind the work that COG has been doing in the context of mutational tracking. So our group at COG, as said in its name, is focusing on the analysis and tracking of SARS-CoV-2 mutations. There are scientists coming from several different academic institutions, from PhD. We are collaborating with other consortia, but I will talk about them better in a few minutes. But that means that COG is playing a crucial role, not only for genome sequencing, but also now interacting with different organizations with the only aim to tackle the SARS-CoV-2 pandemic. So as we heard from David and the previous speakers during this session, SARS-CoV-2 is mutating, and some mutations have the potential to change the fitness of the virus, or to allow the virus mechanism to escape host immunity. So under attention, there are sites known to have immunological roles, either because they are targeted by vaccines or monoclonal antibodies. There are also regions important because they can change, or they change virus infectivity and transmissibility, or because associated with drug resistance. And finally, regions affecting primers, probes, binding sites, which are important in diagnostics. So mutations are accumulating at the population level. And as we know, monoclonal antibodies are currently being trialed for treatment of patients with COVID-19. So if the use of monoclonal antibodies is limited to hospitalized patients, onward transmission of the virus should be very limited. Obviously, assuming effective infection control measures are in place at the hospital. So this could not be the case if monoclonal antibodies are used for prophylaxis, so in the prevention of the disease. So in that situation, there might be evolutionary selection for escape mutations that compromise the activity of monoclonal antibodies. By contrast, vaccines will be taken up by the majority of the population. So in that case, mutations that lead to a reduction in vaccine efficacy will be likely selected for, since they will confer a biological advantage to the virus compared with the wild type. But this scenario is quite complex, and we need to be careful because sets of variants spreading through population with these mutations in reality will depend on a balance of several factors, including whether transmission and infectivity are changed. And lastly, it will be important to carefully characterize potential cases of reinfections, which at the moment seem to be quite limited, in order to identify whether there were mutations present that may be associated with natural immune escape or other factors that might explain the infection itself, for instance, age and comorbidities. So this shows the two different approaches that we are engaging at COG. So from one side here on the left, a data-driven one which focuses on phylogenetic studies, classifying mutations by frequency, the presence or not of homoplasies, the level of entropy, and if there are infection cases or not. On the other side, a targeted approach which employs a surveillance of specific mutations that have already been characterized or epitopes that we know are important for vaccines and monoclonal antibodies. So the last bit down here in this diagram shows how we could further evaluate the selection of the most important mutations through validating experiments engaging other virology groups, which are in partnership with our consortium. So this is about the outputs and how we can actually communicate our findings to other authorities. So in the next few days, a dashboard, a combination of graphs and a database will be available online. So through this interactive tool, we want to share information about the high priority mutations, but at the same time, we want to be able to pick up meaningful mutations as soon as they arise. So these are just some examples of the kind of graphs that we want to use. These, for instance, show the frequency of a list of mutations in color schemes over time. And it's going to be something clickable, if it's possible, where you have access to graphs and visualization, showing, for example, the frequencies over time for each mutation, and other features such as geographical distribution or occurrence metrics, which is important to reveal how different sites might interact. Unfortunately, limited by time, I can't say much on this, but all of our data will have public access and will be stored in a user-friendly database. So COG-UK is a research organization, and it interacts with other consortia, and each of them takes a different angle to study COVID-19. So the Coronavirus Immunology Consortium is looking at the characterization of the immunology relating to viral variants, and they will provide us with a list of epitopes targeted by T cells. So cellular and humoral responses are very important in host immunity, and both should be taken into account if you want to monitor immune escape mutations. Isaric 4C here in the center is more focused on the clinical side. In particular, with them, we are looking for correlations with a different group of individuals to see whether some virus variants can change the severity of the disease, for example. And lastly, the Geno2Pheno Consortium, which will be responsible, once funded, for phenotypic evaluations of specific mutations of interest by using cell lines and animal models as well. So what we do is to give them a list of high priority mutations, which are then further evaluated by scientists with different expertise, as you can imagine. But let's try to the big picture of what's happening. So where do we get the samples? So Test and Trace is responsible of administering test and contact tracing. Most of the logistics is now under Deloitte's responsibility. And at the opposite side, we report to different executive agencies of the Department of Welfare and Social Care, the MHRA here at the bottom, which is interested in our findings on mutations, in particular on the effect of mutations on vaccines. And we are liaising with them to collaborate in post-market surveillance. Then we have also the public health public health agencies, and PHC in particular, that in the context of SARS-CoV-2 mutations, they are responsible of mutational risk assessment in collaboration with NERVTAG, which is one of the government's advisory groups. And here you can see some of them. We heard a lot this year talking about SAGE, but we should consider also the others, like the Joint Biosecurity Center, the JBC, for instance, which provides real-time analysis of infection outbreak and provides policy advice for new circulating variants. And lastly, the JCVI, which provides recommendations on anything concerning vaccination, such as scheduling and vaccine safety. At the end of this long chain, there is the government, which is responsible of making decisions in public health interest. So the entire process, as you can imagine, is quite complex and require a great coordination. So COG-UK's mutational tracking is only one component of an end-to-end process to control these public health challenges. So we all hope that this pandemic will end soon, and we should do our best in order to get prepared, as David said, especially now with vaccines rollout when significant mutations might arise. So here, I just want to acknowledge the underserved people as other people with who are working for COG in the front line and behind the scene. And I would like to acknowledge all of them for the incredible work they are doing. I would have to thank all the scientists that were more directly involved with the mutational analysis group. And I think this is the end of my presentation. Thank you.
Ali, thank you. That was really excellent. It's always hard to finish out a conference session or symposium like this. So thank you very much. I'd just like to thank all of the other speakers in this session and all of the other sessions. I hope you'll all agree with me that it's been a really excellent afternoon. So Sharon, I think you're up next.
Yeah.
For the final comments.
I just asked people to stick with me for 1 minute because that's how long I'm going to be. So I just wanted to, echo the thanks that you just heard from Andy for the chairs and the speakers. I've so enjoyed today. I've been gripped to my chair. It's been 3 hours, more than 3 hours of really great science. It is a marathon, but it was really worth it. I wanted to thank also the organisers. So that's Andy and Kim. So thanks for doing that. And I think that just huge thanks to the consortium. I mean, there's around 300 people. Many of them are volunteers. And we're good together, and I think this is why the consortium is continuing to thrive in the way that it does. So we are planning to put a recording out once we're ready to make sure that it's all good to go. So please do look out for that. And I would just say I hope you've enjoyed it. I hope you have a nice evening, and hopefully we'll be running another of these showcases in the new year when we'll be talking about new work that we've achieved. So thank you again for listening and for your interest, and good night.
Thanks, Sharon.
Good night.
Bye.